أصحاب السعادة السيدات والسادة، أسعد الله صباحكم وأهلا ومرحبا بكم في الندوة العلمية لجائزة الأمير فيصل بن فهد الدولية لبحوث تطوير الرياضة العربية، التي نتشرف أن تقام هذه السنة في دبي، وبالتعاون مع جائزة محمد بن راشد آل مكتوم للإبداع الرياضي. فأهلا ومرحبا بكم في رحاب الإبداع والتطوير، وإن شاء الله نستفيد جميعا مما سيطرح في هذه الندوة من بحوث قيمة تساهم في تطوير الرياضة العربية وتساهم في إضافة الشيء الكثير إلى العاملين في إدارة الرياضة العربية من أعلى مناصب الهرم القيادي إلى أدنى المناصب. فأهلا ومرحبا بكم مرة أخرى. ويسرنا في البداية دعوة الأستاذ محمد القرناس أمين عام جائزة الأمير فيصل بن فهد الدولية لبحوث تطوير الرياضة العربية لإلقاء كلمته، فليتفضل مشكورا.
بسم الله الرحمن الرحيم، والصلاة والسلام على أشرف الأنبياء والمرسلين نبينا محمد وعلى آله وصحبه أجمعين. الأخوة العلماء والباحثين، الأخوة الفائزين بهذه الجائزة، ضيوفنا الكرام، أحييكم أطيب تحية، تحية الإسلام. السلام عليكم ورحمة الله وبركاته. يسرني في هذا اليوم المبارك وفي هذه الندوة الطيبة التي نستهل فيها برنامج حفل توزيع جوائز الفائزين بجائزة الأمير فيصل بن فهد بن عبد العزيز رحمه الله الدولية لبحوث تطوير الرياضة العربية. هذه الندوة التي تأتي على هامش حفلنا لهذه الليلة لتسليم جوائز الفائزين بها. ويسرني في هذه المناسبة أن أرفع أسمى آيات التقدير والعرفان لدولة الإمارات العربية المتحدة لاحتضان حفل تسليم هذه الجائزة وندوتنا هذا اليوم في مدينة دبي، هذه المدينة التي تسابق الزمن وأصبحت أحد أهم المدن في العالم أجمع وملتقى الجميع. أيها الأخوة الحضور، إنما نحن بصدده هذا اليوم من إقامة هذه الندوة العلمية والتي تسلط الضوء على الأبحاث التي تقدمت لنيل هذه الجائزة، إنما تعكس بمشيئة الله ما وصلت إليه هذه الجائزة من مكانة علمية رصينة، وفي النوعية الجيدة من الأبحاث التي تساهم بمشيئة الله في تحقيق الهدف الرئيسي لهذه الجائزة، ألا وهو تطوير الرياضة بشكل عام والرياضة العربية بشكل خاص. هذا الهاجس الذي كان يراود صاحب هذه الجائزة الأمير فيصل بن فهد رحمه الله لإيمانه بأنه لا رياضة بدون مرتكزات علمية، وأن التطور والتقدم الأمثل للرياضة في وطن العربي الكبير لا يتأتى إلا من خلال البحث العلمي والدراسات الرصينة المبنية على العلوم والمعرفة. وما نشهده اليوم من تجمع لنخبة من العلماء والباحثين في مجالات التربية البدنية وعلوم الرياضة في هذه الندوة العلمية إلا دليلا على بلوغ هذه الجائزة لأهدافها. أيها الحفل الكريم، أيها الإخوة الحضور، أحييكم مرة أخرى في دبي، وأشكر لكم حضوركم هذه الندوة، وأخص بالشكر الجزيل للفائزين على تجشمهم حضور استلام هذه الجوائز من مختلف دول العالم. وبسم الله وعلى بركته أعلن عن افتتاح فعاليات هذه الندوة باسم صاحب السمو الملكي الأمير نواف بن فيصل بن فهد بن عبد العزيز، الرئيس العام لرعاية الشباب بالمملكة العربية السعودية ورئيس مجلس أمناء هذه الجائزة، متمنيا لمداولاتكم ومناقشاتكم كل التوفيق والسداد، والسلام عليكم ورحمة الله وبركاته.
شكرا سعادة الأستاذ محمد القرناص الأمين العام لجائزة الأمير فيصل بن فهد الدولية لبحوث تطوير الرياضة العربية على هذه الكلمة القيمة وهذه المشاعر الصادقة، وإن شاء الله بهدف تعميم الفائدة ونقل المعارف إلى قطاعنا الرياضي، فإن هذه الجائزة حرصت على دعوة الفائزين واستعراض بحوثهم وتجاربهم ملخصات عنها أمام العاملين في القطاع الرياضي بهدف الاستفادة وتعميم الاستفادة على الجميع. بعض المحاضرين حيتكلمون باللغة الإنجليزية والبعض باللغة العربية، فاللي يحتاج أجهزة ترجمة موجودة في الخلف باللغتين، من أجل طبعا قبل ما نبدأ في الجلسات فاللي يحتاج بإمكانه أن يتفضل ويستلم جهاز من خلف. الجلسة الأولى ستكون مخصصة للبحث الفائز في المحور الأول، وهو دور العوامل الوراثية في الرياضة. 3 أو 4 من المتحدثين حيكونون موجودين، وسيرأس الجلسة الدكتور محسن سليمان العاملي، خبير البصمة الوراثية والأدلة الجنائية، رئيس ومؤسس مركز التقنيات الحيوية المتقدمة بدبي، فليتفضل لرئاسة الجلسة ودعوة السادة المتحدثين فيها.
بسم الله الرحمن الرحيم، والصلاة والسلام على سيدنا محمد وعلى آله وصحبه أجمعين. لي الشرف الكبير كذا أن أترأس هذه الجلسة في محور مهم جدا له تطبيقات عديدة في مجالات مختلفة، واحد هذه المجالات اللي أنا حقيقة كنت كجزء من تقييم البحوث في هذه الجائزة اللي اتجهت بالرياضة كذا إلى محاولة معرفة التطبيقات الحديثة وفائدتها على الرياضة، خاصة أنه الجين كما يعلم جزء كبير منكم هو الجزء الأساسي لوظيفة الإنسان، وبالتالي التنبؤ بمعرفة إذا كان هناك مقدرة أو استعداد لشخص ما أن يكون رياضيا مميزا. هذه البحوث طبعا أنا أريد أن أدخلكم في الجانب الذي أعمل فيه، هذا الجانب التنبؤ بأمور كثيرة. حقيقة المركز الفائز اللي ستستمعون إلى تفصيل دقيق عن البحوث مركز رائد، لا أقول في مجال الرياضة ولكن في مجالات عديدة جدا. أنا شخصيا كذا مع وجودي في هذا المجال لأكثر من 25 سنة في مجال الجينات الوراثية، طبعا في تطبيقها في الجانب الجنائي أكثر حاجة والجانب البيئي والتنوع الحيوي، أجد على أنه البحوث المقدمة كانت بالنسبة لنا أيضا استفادة كبيرة، خاصة تطبيق التقنيات المتقدمة اللي الآن استطاعت في فترة من الفترات قبل 20 سنة كنا إذا أردنا أن نفحص جين واحد يأخذنا أيام. الآن بالتقنيات الحديثة واللي سيتحدثوا عنها قادرين أنهم يفحصوا في مرة واحدة، يبحثوا عن جزيء من المركب الوراثي 195 ألف موقع، فطفرة كبيرة جدا. سأترك المجال لخبراءنا أن يتحدثوا عنها. You most welcome. I have a pleasure to invite your team, to take the stage, all of you. So please come. فاسمحوا لي كذا بدعوتهم وشكرا جزيلا. سنترك المجال للأسئلة بعد أن يقدم كل أحد منه بحثه وما توصل إليه. شكرا. اسمحوا لي أن أقدم المتحدث الأول اللي هو كلود وشارد. قد يأخذنا وقت طويل لو استعرضنا البحوث اللي تجاوز في هذا المجال. وطبعا من حاصل على الدكتوراه كآخر مؤهل سنة 1977 في مجال البوبوليشن جينيتكس في الوراثة السكانية. من الجوائز اللي حصل عليها the Honor Award from the Canadian Association of Sports Science 1988 موجود في الكتيبات الموجودة عندكم. كما قلت أيضا من الجوائز the Citation Award from the American College of Sports. لا أستطيع أن لو كما قلت لو مشينا في الجوائز التكريم اللي حصل عليه لا أخذت من الجلسة كلها، لكنها كشيء مختصر عنه. طبعا له أبحاث كثيرة، وأكثر الأبحاث اللي كان قدمها طبعا في الجينات المسؤولة عن السمنة على سبيل المثال، وبدأ أبحاثه من فترة طويلة في الجينات التي تتحدث أو لها علاقة بالرياضة والتنوع. هو يعمل في الهيومن جينومكس لابوراتوري في بينغتون بايوميديكال ريسيرش سنتر جامعة لويزيانا، وله كثير من الأبحاث. مشارك أو عضو في مؤتمرات وكثير من الجمعيات العلمية حول العالم كأمريكان كولج اوف سبورتس ميديسين وهذا أمريكان ذا أوبسيتي سوسايتي. فأترك المجال للبروفيسور كلود إنه يبدأ بالتحدث عن الجانب من هذا البحث. لأن please we are invited to take the stage.
في مايك معاه.
Thank you, Mr. Chairman. Thank you for your kind words. And good morning, everyone. It is a pleasure for me and my colleagues, Doctors Sarzyski, Rankinen, and Wolfhard there in the audience, to be here to present some of the findings of the science that we've been able to perform with the support of the Prince Faisal Prize. I will present an overview of the conditions under which we can make progress in the field of defining the genetic and genomic basis of human physical performance. And my colleagues will report more specifically on the research we have performed in the context of the Prince Faisal Prize. So I will briefly speak about these 5 topics. First, a word about human variability in sedentary people, the range of responses to training, the genome-wide search for predictors, what is personalized exercise training, and finally, what are the challenges that we have to meet if progress is to be made in this field. Let me begin with this notion. Not everybody is equal when it comes to performing in events that require a large amount of physical and motor skills. For example, and I'm going to use only examples, but you can, I'm sure, realize the implications that they have. Here we have the result of a study we published many years ago on the skeletal muscle biopsies obtained for more than 400 subjects, young adults who were sedentary, so they were not trained. And we took a biopsy of the vastus lateralis muscle, and here you have the distribution of the proportion of type 1 fibers, the fibers that are oxidative and have endurance as their main characteristics. They are slow twitch, they don't contract very fast, but they can work for a prolonged period of times. And as you can see, the average in this group, the average proportion of type 1 fibers was 46%. That is pretty much in line with what we see in the literature. We assume that about 45 to 50% of the fiber types in the vastus lateralis is type 1. But look at the extent of the human variation here. We have some subjects who have as low as 15% of their fibers as type 1, and others who have 75 and more, a tremendous range. Now, if you are someone who has an interest in endurance performance, and the muscles are going to be solicited in the kind of activities you're going to engage in, and those muscles have a very high proportion of type 1 fibers to begin with, then you are at a selective advantage. And the opposite, of course, if you have a very low proportion of such fiber types. So what we observe, what we see, what we have inherited, even when sedentary, plays a big role in what is accessible to use subsequently on the performance scene. The same is true for maximal oxygen uptake, maximal aerobic power. Here, this is an example of a subset of 174 young adults from the Heritage Family Study. These people were recruited to take part in that study because they were sedentary. And as you can see, in the sedentary state, we have, on average, in young adults, about 40 to 42, 43 sometimes, maximal oxygen uptake. That's the average. But the average is very misleading. We do have some people who only have 25 and 30 milliliter of oxygen per kilogram of body weight per minute, while others can be as high as 50, 55, and even 60. So here again, a tremendous range. And if you are in a young adult or an adolescent who has an interest in endurance performance, you want to be on the far right. I thought I could, yes, I could probably.
Can use that.
Yeah, it doesn't show well. Anyway, you see what I mean. If you are the far ahead of the distribution on the right, you are clearly at a very strong selective advantage if you get engaged in such activities. But this is not randomly distributed. Here, what we're showing is that a low maximal oxygen uptake or high maximal oxygen uptake cluster in families. Here we have about 90 families in the Heritage Family Study, and each family has parents, adults, and offspring also measured as adults. And you have bars, and within each bar you have dots. So one bar represents a family, and you have the dots that are plotted within each bar, and these are the parents and their offspring. And as you can see, the mean, that's the red line, the mean was about 2,500 milliliter of oxygen. But there was a standard deviation of 700 milliliter. And as you can see, some families cluster, all the subjects cluster below the mean, and others at the end, all the subjects in the families are above the mean. So we have aggregation of maximal oxygen uptake. We have family lines with people exhibiting high VO2 max and others low cardiorespiratory fitness. And the irritability coefficient derived from such data attains approximately 50%. Okay, so this is in the sedentary state, just a few illustrations. Now, in addition to the sedentary state, of course, you need to add the effects of training. And here again, we have huge differences in responses. The common view in the past was this. When people exercise, they improve their aerobic power or their performance, and the relationship is more or less linear. The more training you do, the better is your gain in power or in capacity or in endurance. That is a thinking that had to be revised based on a number of studies. And I'm going to show you a couple of them. In this study, for the first time, we began to look at variation in response. We published that in 1984, so exactly 30 years ago. And what we showed here is in 47 young adults that we had trained in the early 1980s, with an identical program for 5 months, we saw that even though the average gain was about 20%, there were some who barely registered any increase, and others who obtained a very large gain in maximal oxygen uptake. So that the gain in maximal oxygen uptake ranged from about 0 to a full liter of increase. Tremendous heterogeneity in the ability to respond to training, to the same stimulus, the same volume of exercise, the same training program. In the Heritage Family Study, which is a much larger study, we also observed the same phenomenon. Here we have an average gain of about 384 milliliter of oxygen, but a standard deviation of 200. And as you can see to the left here, we have plenty of subjects who gain very little and those at this end, on the far right hand, increase their maximal oxygen uptake quite significantly. So this is not exactly how it happens when athletes are training, but we cannot study that in the laboratory in the same way as we can under standardized conditions. And when we standardize the conditions, define the volume of exercise, the intensity of exercise, and then expose a group of people to such a stimulus, we see that some people respond well, others respond average, and others still don't respond. And that's the range that we see here. Now, to understand whether this range of response was in any way associated with family lines, or whether there was a genetic component to it, we started to do repeat the same studies with pairs of identical twins. And this is the first study that we did in this area. And it shows that on the x-axis, here you have the gain in maximal oxygen uptake of one member of a twin pair, and on the y you have the other member of a pair, the brother. So each dot is a pair. Now these twins were exposed to 20 weeks of exercise 4 times a week, very standardized. And as you can see, there were pairs that were in which all the members were high responders, and others were both average. Some were low responders, and if you look down here in the corner, there's a dot, and that's a pair. And here we have both brothers of this pair training for 20 weeks and experiencing no gain in maximal oxygen uptake. Now this is not because they didn't do the program. Compliance here is 100%. They train in the laboratory in front of us, monitored with electrocardiogram all the time. So we know that they did the program. So we had, to summarize that, we had here an F ratio of about 8, which means that there were 8 times more variation between the pairs of twins compared to what we observed between brothers. So that's a strong indication that there are family lines, in this case twin lines, in the ability to respond. We repeated this study 4 times to make sure that we had a true phenomenon, because it meant that the next step would be to go after the genes, and that is a long and expensive process. In one of our studies, we focused more on high intensity and aerobic performance, and same too, here it was 15 weeks of high intensity intermittent training, and here we have a test of a maximal power output over 90 seconds, and you see the same trend. Some pairs were low responders and other pairs were high responders, and we had about 8.5 times more variance between the pairs of twins as opposed to the brothers within each pair. And then in heritage, which again is the largest study, we found the same thing. Here we have the families of Caucasians. We also have African Americans families in the heritage. I'm not going to show these results. So the mean gain was about 400 milliliter. Each bar here represents a family, and as you can see, some families had all their members gaining, but gaining below the mean. And here, families where subjects gain above the mean. Irritability was about 47%. So we gather from these studies that the prior thinking about the relationship between exercise and weight gain, in which some people were endowed at baseline, had a higher VO2 max at baseline, and the thinking was that they maintain that higher endowment level independent of training, such that everybody would respond linearly and additively to the exercise program. That model does not work. Rather, we need to go to a model like this, where there are differences in the response to training that are conditioned by your genes. And now the challenge becomes which genes. And these are the studies that we have done to address this. Ideally, you would like to do a complete study with the same people, training them at one level, and then a year later continuing with another level, and then another level longitudinally, the same people. That, of course, has proven not to be doable, but what we have are studies in which the program was of low intensity, higher, even higher, very intense, and across the whole range of exercise training, intensity and volumes, and we have always observed the same phenomenon of a genetically conditioned ability to respond to the training program. Now, this is performance. Keep in mind, though, that this is also the same thing for health-related indicators. Like, for example, here, this is fasting, insulin and insulin sensitivity. In the Heritage program, based on hundreds of subjects, we have seen that the fasting insulin is decreased by 2%. That's what the textbook would predict. If you exercise, you need less insulin to maintain your blood glucose. And your insulin sensitivity was increased, and here it did increase by 10%. That's the average. But when you look into the details of it, and here I'm showing you only fasting insulin. So we have the subjects here, and we have the changes. The mean decrease in fasting insulin was about 7 picomole, but the standard deviation was 32, very large. Now what we have here are the people who registered a decrease. Those in green truly improved their fasting insulin. Then we have some who were probably within the area of measurement. They tended to decrease or increase, but we have the red group here, very frank subset of individuals that experience a higher fasting insulin as opposed to a decrease in fasting insulin. So they go against the textbook. They have what we have called an adverse response to exercise. And that is something that is of importance when we look at what is necessary to provide a foundation for personalized exercise medicine. You want to be able to recognize these people and to find another way to help them, because exercise is not necessarily good for them. Okay, so and we have seen this not only for fasting insulin, but everywhere we have looked. Systolic blood pressure, we have people who do not respond well. A lot of people reduce their blood pressure, but some increase their blood pressure. HDL cholesterol. Some people improve their HDL cholesterol, but there are some others who experience a decrease in the good cholesterol with regular exercise. Triglycerides and fasting insulin, the same thing. It's pervasive. Now, I'm not going to continue talking about exercise and health, but keep in mind that what we're saying about performance also is relevant to exercise and health as well. Now, how do we go about finding the genes and the DNA variants that are of importance and account for this extraordinary degree of human heterogeneity or degree of human adaptability to exercise? Well, of course, this was greatly facilitated by the advent of the human genome sequence, which was published about 12 years ago now, and by the work done on the extensiveness of human variation in the sequence of the genome. Two big projects the HapMap project, and the 1,000 Genome projects. These large international efforts have given us the tools to begin to investigate the foundation of the human heterogeneity in adaptation to exercise. This is the model that we use in our laboratory to frame the discussion. When people exercise, they improve their fitness. And when they improve their fitness, they tend to be healthier or to be able to achieve a higher degree of performance. There are also some traits where you exercise and you improve health and performance without changing fitness. For example, when you exercise, you have a honeymoon period during which you dispose of glucose and your sensitivity to insulin is elevated for a few hours. That occurs without any change in fitness. So, but what we are interested is in the role of the genes. And by that we mean the role of DNA sequence differences in and around the genes. It's not just what are the genes involved, but we want to know which genes exhibit DNA sequence differences that relate to this extraordinary degree of human heterogeneity. And there are efforts, and we are involved in such efforts as well, to try to understand how these DNA differences impact on the ability or on the interest to exercise, on fitness in the sedentary state, or on your health and performance status even when you are in the untrained state. But we are even more interested into these two additional arrows, the ones that are in the interface of the exercise and fitness, and of fitness and health and performance, because this is where the modulation of the benefits are taking place. We have noticed in the data I just showed you that when people exercise, there are a large degree of differences in fitness. Well, that is what we are trying to do here. Dissect out the fundamental genomic reasons why this occurs. So that's our model. We will be talking mainly today about these arrows here in the rest of my presentation, and in those of Dr. Rankinen and Sarzinski. So a couple of years ago, we published the first genomic screen of the ability to respond to exercise using maximal oxygen uptake as our criteria. And we genotyped about 300,000 markers across the genome. And here we have an overview of the findings. Here you have the various chromosomes, and on these bars you have the results. This is packed with p-values. Here on chromosome 1 we have tens of thousands of SNPs that were investigated, and the p-values are there, and you can see that there are some p-values that are way above the others, while down at the bottom the mass of them is aggregated there. We call this a Manhattan plot. And then, so we can see that across the genome there are a few SNPs here, a few markers that rise above the surface, and those are the ones that we are mostly interested in. And when we look at the most important SNPs in the surveys that we did, we found that there were 21 of those markers that when analyzed together accounted for about the degree of irritability. So I told you before that the irritability of the ability to respond to exercise was about 45 to 47%, and with these 21 SNPs we account for all of this, 47%. So we use those, since there are 21 SNPs, at each it's a change in one DNA base. We have therefore two version. One comes from our father and the other one from our mother. So we calculated at each of these 21 SNPs the number of good ones, the number of alleles that a person had that was associated with higher gain in fitness, and we summed this across the 21 SNP. So that the range was from 0, someone who had absolutely no good SNPs, to 42, someone who had only good SNPs inherited from both the mother and the father, and had therefore 42 good alleles. In practice, we did not have individuals at these extremes, but we had some subjects who had less than 9, and another group who had more than 19. And as you can see, those who have few good alleles increase their maximal oxygen uptake by about 200 milliliter of oxygen. And those who had more than 19 increase their maximal oxygen uptake by 600. A threefold range, just on the basis of this simple study. Now, the sample size is too small. If we had 10,000 people, we probably would have had some who had less than 5, maybe less than 2 good alleles, and others who had more than 20, 25, 30 good alleles, and we think that the spread will have been even greater. But despite this limitation, we still find that we can predict a threefold range in gain with these 21 SNPs. And these SNPs are in genes or near genes that we believe make sense. They are enriched in developmental pathways, including the pathways that relate to the regulation of gene expression, the formation of new blood vessel, skeletal and cardiac muscle growth, and also genes that regulate cell death and apoptosis. Okay. Another very nice paper was published by my colleague, Dr. Rankinen, and it related to, this time not maximal oxygen uptake, but an indicator of some maximal exercise capacity. The changes in submaximal exercise heart rate. Let me explain that to you. So when we exercise, we increase our power output progressively. We have a corresponding increase in heart rate. For example, this is what we have. You increase the power output, we have a higher heart rate before training. Then after 20 weeks of training in heritage, you would expect that at each of these power output, the heart rate will be lower, 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 lower. So we took at one level, at 50 watts here. At 50 watts, we asked the question, is there a difference before and after training? And are there familial aggregation for such differences? And if so, can we explain human variation in the heart rate response by key genes and DNA variants? This is the distribution of the changes in exercise heart rate at 50 watts. And as you can see, there was a decrease on average of 11 beats per minute, but a standard deviation of about 10. There was an irritability coefficient of 34%. So you can see here, most people decrease their heart rate. But the some, after 20 weeks of exercise, when no problem with compliance, they did all the session, they exercised, they worsen. They have a higher heart rate rather than a lower heart rate. So what we did, we explored this with our panel of genomic markers, and true enough, with 9 SNPs, we were able to account for the whole irritability, 34%. And we took that a little bit further, did the same exercise as before, summed the number of good SNPs across the 9 SNPs, with the theoretical maximal score would be 18 in this case. And as you can see, those who had 16 good SNPs, 16 good alleles associated with improvement had on average 20 beats per minute less when we compare before and after training, while those who had only 9 or 10 of those SNPs had no change. Tremendous differences in the ability to be more efficient at the sub-maximal level. So what I told you applies not only to the maximum, but also to some maximal power output and related cardiovascular adaptation. Now, just to see how it works, I have color-coded. So those who are at this end, in red, bad response, and green, very good response, excellent response in green. And then we plotted the whole population of about 450 subjects to see whether we could predict if a red was responding bad and the green responding well. And as you can see, most of the red are concentrated here. We did pretty well. And the green, those who really improved, are concentrated here. So the prediction appears to be working. It's not perfect because the sample size is too small to have an even bigger spread of the scores. But we're doing a good job just with the SNPs. And don't forget, the irritability is only 34%. It's not 100%. So we cannot expect to be able to classify on the basis of DNA variants 100% of the people, because there's still 2/3 of the variance is not related to the genes. Okay. Now, what does that mean for exercise training and training for performance? I would like to use endurance performance as an example, but endurance performance cannot be easily studied in a real situation of endurance performance, because you would need to have people at your disposal for long periods of times and under standardized conditions. So for laboratory studies, we often rely on maximal oxygen uptake because this is a strong correlate of endurance performance. And actually a number of studies have been done on this, and the correlations are ranging from 0.6 to 0.9. But it's maximal oxygen uptake is only one of multiple determinants of any types of endurance performance. It's a strong one, but there's a lot of other things coming into play, and that needs to be recognized. In addition, maximal oxygen uptake is a very complex physiological trait that is the aggregate of a lot of things. It's the aggregate of oxygen transport, oxygen transfer, oxygen utilization, and then the heart, the blood vessel, and then the muscle machinery. All of this is involved. So when we talk about, just from a biological point of view, when we talk about focusing on this, we are far from where the action is in the heart or in the oxygen transporting capacity, or the fiber types and the protein concentration in the right tissues. So let's keep that in mind. It would be easier to find the genes if we could work at that level, but it has proven not to be feasible in large groups of people. Okay. So if our goal is to develop a world-class endurance athlete, I think we will submit that it begins with someone who has a very high VO2 max in the sedentary state. If we go back to our curve here of a VO2 max in the sedentary state, we know that there are a few people here who have more than 50. Those will be good candidates, because to be able to perform on the world stage and have a chance to be on the podium, if there's a strong endurance component to an event, you need to be in the range of about 80 milliliter of oxygen. You need to reach that, about 80. If you don't, you might make it, you might make the national team with 75, but you're not going to be on the Olympic podium. And here, 1 in 50 people has that type of maximal oxygen uptake in the sedentary state, approximately 1 in 50. Now, in addition, you must be one who has a high ability to respond. You must be highly trainable, and to reach this value of about 80 milliliter. For example, the training range in the response of 50 to 60% to a given volume of exercise, in our opinion, is not very frequent. Heritage shows us that it's not very frequent. If we, again, take a subgroup of young adult males in Heritage, the range was from 0 to about 60%. So that's the group here that we're talking about, a very low prevalence. But if you're there and you started with a high value of around 50, you have a good chance of being able to make it to a high level of 80 milliliter per kg per minute. Keep in mind that there is no correlation between baseline and trainability. This is based on the Heritage data. I think there's about 600 subjects in there, and the correlation is 0.08. There's no relationship. You can have a low baseline and a high trainability, and a high baseline and a low trainability. So what we are after now is the joint occurrence of both, high and high. And based on all of our training studies, we estimate that it is about between 1 in 1,000 and a few in 10,000. 1 in 1,000 to 3 to 4 in 10,000 would have both genotype, endowed in the sedentary state and endowed for trainability. So it does exist in population, but it's not very common. Okay, so how to go there? Well, when it comes to the sedentary state, normally you can measure it. So there's no need to have sophisticated genomic predictors for it. You just measure it in the lab. What you don't know is how high can this person go without challenging the person and measuring it at that time. So genomic predictor may come handy here for the trainability. You don't necessarily need them here in the sedentary state. But developing a genomic diagnostic for high trainability is a major undertaking, because there is a risk of generating results that are false, and therefore you will be providing information that will be misguided for the participant. So these are the challenges that we have to meet. We need diagnostic in exercise medicine to identify talent for specific activities or families of activities. We want to be able to identify good and excellent responders. We would love to be identified to non-responders and adverse responders so that we could advise them appropriately. We need to potentially identify adverse reactions, for example, adverse drug-exercise interactions. And we want to be able to customize exercise recommendations for health and exercise training recommendations for athletic training. But all of this require powerful and precise diagnostic tools. What is a genomic diagnostic in this context? Well, it means that we have a DNA variants that modulate to the maximum of the irritability, the response to training exposure. And these DNA variants must have sufficiently large effect size on the trait of interest, for example, delta VO2 max. It must have high sensitivity and specificity, and it must be carrying sufficient information to have a positive rock curve. Let me show you what it means. But first, let's define sensitivity and specificity. And this is absolutely critical. The sensitivity of a diagnostic is a proportion of subjects with the trait of interest, in this case, say, high trainability for VO2 max or muscular strength or muscular power, is the proportion of subjects with the trait of interest who test positive when classified by the diagnostic. So they are the real positive. And the specificity is the proportion of subjects without the trait who test negative, the true negative. So that's important, because if your test does not pick up the positive and picks up a lot of the negative cases, you're not discriminating properly. Or in contrast, if you leave out a lot of positive, you'll not be able to give good advice. So it's very demanding to have a good diagnostic. We evaluate this with this tool, the receiver operator characteristic curve, or ROC curve. And here we plot the true positive as a function of the specificity. So the sensitivity is on the y-axis, and the 1 minus the specificity, or in this case 100 minus the specificity, is on the x-axis. This line means that you cannot predict. It's a toss of the coin, 50%. You have a chance of being right if you say yes, you are gifted, or no, you're not gifted. It's absolutely no power. But if you add a diagnostic that will give you a curve of this type, then you would have quite a bit of power, because you have here very high sensitivity with no downside, with no negative cases. And ideally, you would like to be here. When you would say here to a young, promising athlete, or someone who wants to become one, that he has the right genotype to be highly trainable, you would be right. And you would not be leaving behind people who have the trait but are not picked up by your test. Now, this is measured typically by the area under the curve, and there are several curves. This one is useless. If you have a curve like this for your diagnostic, it's useless. It's almost equivalent to a toss of the coin. If you move there, you begin to see a little bit of value, but it's not precise enough to, you're playing with the life of people here when you say, yes, you're endowed. You should get into these sports because you have what it takes. That's not powerful enough. If you have that, bingo. You have the strength of a diagnostic to be able to do it. So it's very demanding. So we are in the field where people have, over the last 20 years, have used single genes, ACTN3 or the ACE gene. And they've said that there are commercial entities offering tests here on the basis of one gene. They tell people you are endowed for power performance or for endurance event. This is totally ridiculous. With dozens and dozens of genes, we do not even reach that level here. How can we do that with a single gene? You're doing so much harm with this. Okay, now to reach powerful diagnostic, the conditions need to be right. And here we have two strikes against us to begin with. We're dealing with genomic markers that have small effect sizes, and the sample size requirements to do these studies are enormous. And I'm going to finish around this topic. Typically, a genomic variant as only a small effect size. And that's pretty common for everything we are interested in, because if there were variants with large effect sizes, it will not really be compatible with homeostasis, with life as we know it. We have rather lots of DNA variants in our genome, and they all are responsible for small differences. And that is compatible with homeostasis, with the biology being stable. So when we have a diagnostic that has an increase in prediction of 10% over just random selection, that's not very big. So the diagnostic must be extremely powerful, even at a 10% contribution, to be of value. But to reach that, let's say here we have the case where we have classified athletes and non-athletes, and we have a 10% gain, 1.1 odds ratio, a 10% gain over randomness. Well, to be able to reach a conclusion of the true value of the diagnostic, assuming that, say, the gene that you are interested in as a variant, which is prevalent at about 10% in the population, you will need about 49,000 subjects to establish the validity of the diagnostic. If your diagnostic improves your predictability by 20% for the same 10% allele frequency, you need about 13,000 subjects to reach the conditions under which you will develop the tool. Of course, if you improve the predictability by 100%, you can do it only with about 700 subjects. But that is not going to happen in genomic research. So you need to be all aware that studies with small sample size are doomed to fail. That's the bottom line. Okay, so I go back to the model that I put before, and now I have enlarged it a little bit. We are interested in the genes, but to really tease out all the underlying biology that predisposes someone to be able to compete at the international level, elite level, we will need all the powers of the omics, the sequence variants, the copy numbers, the epigenetic markers, but also we will need the power of the other omics, the transcriptomics, the proteomics, the metabolomics, all the dimensions of the characteristics of the cells and tissues in order to reach more powerful diagnostic, especially as we try to refine our systems of classifications. So there's a lot of work that needs to be done, and we're beginning to have a foundation, but most of it lies ahead. And those of you who are interested in a career in science now, exercise sciences, exercise genomics is a good place to be. Thank you very much for your attention.
Thank you, Paul. تفاصيل الإحصائية مثل هذه النتائج، لكن استخدامات الإحصاء عادة تستخدم لتوثيق تأكد من دقة المعلومات والنتائج اللي توصل لها. فالمتحدث الثاني دكتور سارزينكي, I hope that is an instructor research with also the Human Genomics Laboratory, and he's a member of
sports and medicine is one of the, of course, the team who, did or contributed to this research. And I don't want to go into more details because your book later you have اللي موجود عندكم كذا فيه تفاصيل عن الخلفية كذا. سنترك الأسئلة إذا في أي استفسارات حتى لو كانت بأي لغة من اللغات كذا، نحاول بقدر الإمكان أن نوصلها إلى المتحدثين للاستفادة منها بعد انتهاء المحاضرة الثالثة.
All right, thank you for the introduction, and I want to thank the Prince Faisal Prize Foundation for having us here. We're really honored. On this project, and I want to especially recognize the contribution of Dr. Bernd Wolfart, who is sitting in the audience due to his duties as team doctor for Germany in the Sochi Olympics. His travel plans just got him here this morning, so he contributed a lot and played a large role to the data that I'll present to you today. So based on all of the information that Dr. Bouchard just gave you, as you could imagine, once the field determined that genes were playing a large role in the response to exercise, in performance, many people became interested in looking at the differences between elite athletes and regular everyday people, and specifically looking for genes that might be involved with elite athlete status. So today I'll give you a little overview of some of our previous work on candidate genes involved or associated with elite athlete status, and a little overview of the field as a whole. So before I begin, I should give you a little background on candidate gene studies and the candidate gene approach. So many studies have been designed with the main goal of examining and detecting an association between DNA sequence variation with complex traits. So the researchers want to know, do certain sequence variations within the genome, are they associated with complex traits? Traditionally, these studies use the candidate gene approach. So these candidate genes are selected based on their known biological, physiological, or functional relevance to the trait of interest. So as you could imagine, the success of the candidate gene approach then depends on the correct choice of which genes or which pathways to study. A lot of these candidate gene approach studies use the case control method. So these candidate gene studies take cases who have the trait of interest, controls who do not, and look for differences in the frequency of genes or alleles between the cases and controls. So these studies are trying to find, the answer is one allele of a candidate gene variant more frequently seen in subjects with the trait of interest than in subjects without the trait of interest? So here's a hypothetical example of a case control study looking for the association between a genomic marker of interest and, let's say, a performance trait or elite athlete status. So here on the left, we have our cases or athletes, so they have high performance. And here on the right, we have controls, or those individuals with low performance. And here we're looking at the hypothetical gene X. Gene X has allele 1, which is indicated in red here, and allele 2. So as you can see in this hypothetical example, all of the athletes possess allele 1, whereas none of the controls possess allele 1. On the other hand, all of the controls possess allele 2, whereas none of the athletes or cases possess allele 2. So in this case, we have a perfect association. All of the athletes have one marker of interest, whereas the controls do not. But as we learned from Dr. Bouchard's talk, this is not the case in practice. Many traits are heterogeneous. So here we have the same example of cases on the left, controls on the right, the same hypothetical gene X. And now you can see that most of the athletes have the red allele 1, whereas a couple do not. Whereas the controls here, most of them do not have allele 1, but a couple do. So although this is not a perfect association in that all of the athletes have allele 1, this is still a highly significant association between our candidate marker of interest and the trait. I don't need to go into a lot of detail on the genetics of performance traits, as Dr. Bouchard explained a lot of this for us. So suffice it to say that we know there's very strong evidence that there is a genetic component not only in our inherent ability to have high aerobic capacity, but also our ability or trainability of VO2 max or aerobic capacity. And as Dr. Bouchard showed us, the heritability for VO2 max in both the sedentary state and the trainability of VO2 max is around 50%. However, although these heritability numbers are known, very little is known about the specific genes and gene mutations accounting for these heritability levels. So, as you can imagine, many people are interested in this topic and looking at genes associated with endurance performance traits, and several studies have taken place. And the candidate gene approach has been applied to the endurance performance phenotypes. And through these studies, multiple genes have been reported to be associated with endurance performance phenotypes. And over the past 14 or 15 years, our group has produced a yearly report summarizing the association studies between endurance performance traits and candidate genes of interest. This report started in 2001 and was named the Human Gene Map for Performance and Health-Related Fitness Phenotypes. This was a report that was published in the Medicine, Science, and Sports Exercise Journal, the first issue being in 2001. And the goal of this exercise was to make a human gene map for fitness and performance phenotypes by reviewing all of the genetic loci and markers shown to be related to physical performance or health-related phenotypes in at least one study. So in this first report here, what the authors did was take all of the previous studies examining the association between genes and endurance, performance, or health-related fitness phenotypes and summarize them in one report. And then in subsequent years, that year report summarized all of the evidence for the previous year. So we published 7 of these reports, with the last one being published in 2009, which covered all of the reports up to 2007. And what this shows is all of the chromosomes listed here, including the X chromosome and the Y chromosome, and it shows the candidate genes that had been identified to be associated in at least one study with endurance performance or health-related traits. And you can see that at least one candidate gene had been identified across all of the chromosomes. And so the latest report in total showed about 214 autosomal and 7 X chromosome gene entries and quantitative trait loci associated with these endurance performance traits. And in this last report, we also summarized all of the different papers and different loci for each of the different traits of interest. So what this table shows here are all the different traits that we examined, the number of papers published up to that date for that trait, and the number of gene markers associated with that trait. So you can see here for endurance traits, in 2009, 72 papers had been up to 2009, 72 papers have been published that examined 47 different genes that were associated with endurance performance-related traits. And you can see here for the various other traits, such as exercise intolerance or physical activity, we had 9 papers, 18 loci. But it should be noted that a majority of the genes that we saw that have been identified across the years were based mostly on one positive finding. So one study had shown this gene of interest was associated with one of these performance or endurance-related traits. And on top of that, very few studies had looked at the association of the response to training and some of these candidate genes. In fact, the only studies that showed candidate genes associated with train responses in more than one paper, we had 2 genes that were shown to be associated with train response in more than one paper, and that was the ACE gene, angiotensin converting enzyme, and the apolipoprotein E gene. So thus in 2010, we decided to change the format of our review. Instead of including all of the studies up to that date, we decided to focus only on the studies with the highest quality. So in 2010, we changed the format, and instead of being the human gene map, it is now titled Advances in Exercise, Fitness, and Performance Genomics. So the goal of this yearly paper is to publish a yearly review of the scientifically strongest and most important articles in exercise genomics. And just this year, we all published the 4th version of this. So based on all of our annual reports over the past 15 years, we conducted a literature search of all of the case control studies that looked at the association of genetic markers with elite athlete status. So the data that I will be showing you today is a review of the literature up to about the end of 2012. So one of the first case control studies to really systematically look at the genomic differences between elite athletes and non-athlete controls was the Gene Athlete study, which was led by Dr. Bouchard. And in the Gene Athlete study, we examined single nucleotide polymorphisms, or SNPs, in several candidate genes and found some of them to be associated with elite athlete status, but none have really provided strong evidence of the difference of frequency in alleles or genotypes between athletes and controls. And I'll show you this more in some of the following tables. So here's a brief overview of the candidate genes that have been tested in the Gene Athlete study. You can see the genes are listed in the left. There's about 8 of them. These genes are found in pathways to be associated with blood pressure regulation, muscle contraction, vasodilation, etc. You can see that across these genes, we have examined the association of several markers, which are listed in the SNP column there. And what we were doing in these studies is examining the frequency of alleles between the elite athletes and between the controls. And what you can see here in the p-value column, sorry my laser's not working, NS stands for not significant. So the association was greater than a p-value of 0.05. So you can see that for most of the markers, we did not find a significant association between the candidate gene marker of interest and elite athlete status. For 3 of them, we found p-values less than 0.05. However, for a majority, there is no association. So to give you a specific example of how one of these results tables looks, here's a table looking at 2 of the most well-studied candidate genes in exercise genomics, the angiotensin converting enzyme, or ACE, insertion deletion polymorphism, and the actinin 3 R557X mutation. Numerous studies have examined the association of these 2 candidate genes with various traits such as endurance performance, elite athlete status. And what this table shows is the association between allele frequency between the athletes, the elite athletes in gene athlete, and the controls. So for example, on the top for the ACE insertion deletion polymorphism, we had 192 elite athletes, 189 controls. And you can see that the frequency of the II genotype was not different between athletes and controls. They both had around 20 to 27%. The frequency of the ID and DD genotype also did not differ between the elite athletes and controls. If we looked at this only in terms of alleles, the frequency of the I allele and the D allele between athletes and controls also was not different. And this is the same if we looked at the actinin 3R557X mutation. There's no differences between genotype frequencies between the elite athletes and controls. So at least in terms of the gene athlete study and the candidate genes that we selected, there were no strong associations between elite athlete status. So as we looked at the literature, in addition to the gene athlete study, there are numerous studies examining genetic associations with elite athlete status. Overall, we identified 48 publications looking at associations between various gene markers and elite athlete status. 34 of these studies reported at least one significant finding, significant being defined as a p-value less than or equal to 0.05. And these 34 studies involve 12 candidate genes and several mitochondrial DNA haplogroups or polymorphisms. The remaining 14 papers reported only negative findings, similar to what many of our gene athlete reports showed. So in total, from these 48 publications, a total of 17 candidate genes were examined. The most commonly studied genes were the ACE gene, which I described to you in the previous slides. 14 studies looked at the ACE gene. 7 studies examined the actinin 3 gene, and 4 studies the PPAR gamma 1 alpha gene. So there are some limitations with the previous case control studies looking at the association between genetic markers and elite athlete status. One is that a lot of these case control studies had very small sample sizes, as you can imagine. To accumulate a lot of elite athletes is a very difficult process. And another thing a lot of these studies had was the lack of a very well-defined physiological phenotype. So they might have just said, here are elite athletes, here are controls. There are many different ways to define elite athlete status. It could be being a member of the national team. It could be based on time trials, performance. In the gene athlete study, we based it both on being the member of a national international team as well as having a high VO2 max. So it had a physiological component to it. Another thing I should mention about the majority of the previous case control studies is that a majority of them, the primary hypothesis the study was designed to test, those results were negative, meaning the overall study may have been designed to test the association of one candidate gene between elite athletes and controls in the total population. However, the positive findings they report were actually found only in males or females or male rowers. So their positive findings were not a part of the main hypothesis. And furthermore, they did not correct for the multiple testing. So if they found the findings in males or females, they need to correct for the fact that they ran multiple tests to get those results. And then lastly, and importantly, few of these results have been replicated in further studies, and in particular in larger replication studies. Thus, to date, no strong evidence exists for many of these candidate gene loci. So the purpose of the study that I'll be presenting to you was to use SNP data from the cardiometabolic chip, which includes genomic markers across the genome, to test whether there are associations between the SNPs from these candidate gene loci previously identified and elite endurance status in the gene athlete study. So the methods of our study involved the gene athlete study. I'll give you a brief overview of this study. So the gene athlete study employed a case control design. We had 635 individuals total. Of these, 315 were cases, and cases were defined by two measures. One, to be a case, you had to be a national or international level endurance athlete, and you had to have a measured high VO2 max, with high VO2 max being defined as greater than or equal to 75 mLs per kg per minute. And of our 315 elite athletes, they came from the sports shown here. So we had 105 that were cross-country skiers, 86 biathlon, also athletes from cycling, running, and other sports. We had 320 controls. These controls were sedentary non-athletes with measured VO2 max less than 50 milliliters per kilogram per minute. And all of our cases and controls were matched for gender. We only included males, Caucasian males, and they were matched by geographical location. So here in the last bullet point, you can see the countries where our athletes and controls came from. We had 187 athletes from Germany. We had 78 from North America and about 50 from Finland. So what we did next was, based on the literature search I showed you in the previous slides, I showed that 48 studies had looked at about 17 candidate genes. The cardiometabolic chip includes over 100,000 SNPs, so we perused all of those SNPs and genes from the cardiometabolic chip to see if markers in those 17 genes were available. And in total, we found that 13 genes shown here in this table were available in the cardiometabolic chip with multiple SNPs within many of these genes. So in total, we were able to test 13 candidate genes and 161 SNPs within these candidate genes in the gene athlete cohort. So our genotyping was performed using Illumina and the cardiometabolic chip, as I previously mentioned, in our 315 athletes and 320 controls. The cardiometabolic chip as a whole included over 145,000 SNPs. And for our statistical analysis, we wanted to look at differences in allele frequencies between the elite athletes and the controls using chi-squared test. And again, I said we're looking at 13 loci and 161 SNPs were tested. So thus, statistical significance was set at a p-value less than or equal to 3 times 10 to the minus 4 to control for the multiple testing. So now on to the results of our analysis. So here is a basic table showing the characteristics of the gene athlete cohort as a whole. You can see here that between our athletes and controls, they're about the same height on average, with the controls being heavier than the athletes. And by design, the athletes had significantly higher VO2 max on average, with an average of 79 milliliters per kilogram per minute, whereas the controls had an average of 40 milliliters per kilogram per minute for VO2 max. And again, we can see the distribution of the countries where the cases and controls came from. So here are the results for the analysis looking at the association between markers in the candidate genes and differences in allele frequency between the athletes and controls. So this table here shows are all the associations that had a significance value of p less than 0.10 or lower. And we can see here that about 8 SNPs were associated at that level, with only 7 associated at p-value of 0.05 or less. And of these associated at a nominal value of 0.05 or less, 3 were located in the PPAR gamma coalpha 1A gene, and 3 in the nitric oxide synthase 3. And you can see on these p-values, though, our best p-value was 0.0008, with most of them higher than that. And as you recall, to be statistically significant, our multiple testing threshold for significance was a p-value of 0.0003. So none of these SNPs are statistically significant after taking into account multiple testing. And as you can imagine, the association for the remaining 150 plus SNPs was even weaker, with the association being greater than 0.10. Additionally, we also ran these analyses looking only at the elite athletes with the very high VO2 max. And for this, we defined a high VO2 max as a VO2 max of greater than or equal to 78 milliliters per kilogram per minute, with 78 being the median value for our athletes. So in total, we had 168 athletes with a VO2 max of 78 or greater. We took these 168, compared them to controls across all 161 SNPs, and we had similar findings in that none of the SNPs were statistically significantly associated with elite athlete status. So overall, either in the total cohort or only focusing on those with high VO2 max, none of the SNPs were associated with elite athlete status. So as I alluded to earlier, there are several limitations of the case control design and the candidate gene approach. One is that the candidate gene approach is limited by our current understanding of the biology of the trait. So you only can select genes that are known to be involved with pathways involved with endurance performance, for example. And we're also limited by the assumptions of the function of the gene. So we're only testing gene regions that are known to be associated with our traits of interest, but obviously then we will miss the influence of any other causative variants that are located outside of the regions we tested. And as Dr. Bouchard mentioned in his talk, these are very, very complex traits. Elite athlete status involves many different factors. Psychosocial factors, like some of our presenters later on today will talk about. We have environmental factors, genetic factors, etc. And the genes that are associated with elite athlete status are likely to each contribute a small amount, as Dr. Bouchard mentioned. Thus, identifying the independent contribution of one marker will be very difficult unless we have large sample sizes, like Dr. Bouchard alluded to, in the thousands, which is very difficult when we're talking about elite athletes. And obviously then, most studies generally have low statistical power, as we're not able to get these large sample sizes. Other concerns or limitations of the candidate gene approach is obviously that we have a biased target. These candidate genes are typically selected based on the preference of the researcher, maybe what pathway, what genes that are really popular at the time. And as we know, these are very complex traits. There's heterogeneity between individuals for these traits, especially for elite athlete status. We have a lack of replication or inadequate replication. We don't have large sample sizes that we can use to replicate many of these findings. And one important part that is often overlooked is that negative studies are rarely published. So yes, there are several candidate gene papers published. I showed examples of about 48. However, maybe there's many more negative association studies that were not published. So there's a very strong publication bias in the current literature. So many of these limitations are reasons why we have not found replication in candidate gene studies. One obvious potential reason for non-replication is variation in study design. So several studies have been performed examining the association between candidate genes and elite athlete status. However, they may have used different athletes. So some used rowers, some used runners, some used a combination. We also have differences in the ethnicities examined in these studies, gender, etc. And many of these studies were not originally designed to examine genetic factors. They may have just been epidemiological studies and then within that large epidemiological study, they picked out the elite athletes. An obvious issue is confounding and misclassification in these studies. The broader we define elite athlete status, the more potential we have for confounding and misclassification. In Gene Athlete, we measured VO2 max, so hopefully we help to lessen some of the misclassification and confounding. And one obvious issue is the lack of evidence. Many researchers study these candidate genes without major supporting evidence from the literature. They find one or two studies, they decide to study it in their cohort because it's popular at the time. It could also be the case that the marker that was tested is not a functional marker, or it's not in linkage disequilibrium with functional markers. And then the most obvious reason is that no association actually exists for the candidate gene being tested. So in conclusion, in our study, we found no significant associations between the literature-derived candidate genes for endurance performance and elite athlete status. Thus, it's very unlikely that these loci are contributing to the potential of becoming an elite endurance athlete. It's likely that the sequence variation in endurance performance candidate genes only explain a small percentage, if any, of the total genetic variance in elite athlete endurance status. Thus, there is an overall need for a systematic genome-wide approach in the search for the genetic component of elite endurance athlete status. And this leads nicely to some of the work we did with the gene athlete study that Tuomo Rankinen will present, because it's obvious that the candidate gene approach has not been very successful to date. Thank you very much.
العلمية في هذا المجال كما قلنا يتحدث عن كيفية أو إمكانية التنبؤ باستخدام النتائج العلمية لهذا البحث. Please.
Thank you very much, and good morning on my behalf as well. So my task today is to finally present you the results we obtained from our studies using the generous award from Prince Faisal Foundation. And I will start first by giving a brief overview of the history of scientific studies on sports and genetics. And then I will address with a few slides what I think are the key reasons why we haven't made as much progress in sports genetics as we have in human genetics in general over the last 10-15 years. Then I will briefly outline the methods and design we used to do our study, show you the results, and then address a few discussion points afterwards. So the history of sports genetics goes actually back to the mid-1950s, when a group of Italian scientists published first studies using twin study design to address a question whether sports selection aggregates or differs between monozygotic and dizygotic twins. And already in those studies, they provided evidence that the monozygotic twins, who are 100% genetically identical, are more alike amongst themselves in selection of the sports of their choice than dizygotic twins, who share only 50% of their genome identical by descent. Then these studies were followed by actually two efforts to study athletes in Olympic Games. In Summer Olympics 1968 in Mexico, and then 1976 in Montreal. the scientists collected samples from athletes and basically compared common blood genetic markers between athletes and non-athletes. And that was the first attempt to really look at the molecular level, the genetic differences between athletes and non-athletes. These studies were done before the DNA era, before we had the tools to actually study and amplify DNA from our cells and to address these questions at the individual nucleotide level. And as Dr. Sarasinski already mentioned, the first systematic case control design to really study elite endurance athlete status was launched in the early 90s by Dr. Bouchard. And actually the results I present today are a result of that original effort. And we've had several other attempts to test the differences between athletes and non-athletes in terms of DNA sequence variation in the 2000s over the last 15 years. And altogether, we have over 100 reports dealing either with athletes or with maximal aerobic capacity, but we haven't, and all these studies have used the candidate gene approach that we just heard of, but we haven't really found nothing convincing yet, which is actually surprising given all the success we have in complex genomics with traits like diabetes or heart disease and so forth. So I think we have two major hurdles in our research approach that has slowed our progress down. And first one is the selection of candidate genes and the DNA sequence variants for the candidate gene studies. And that is a very, very difficult task, because now I need to emphasize that the target genes that we have selected, even though the approach has failed, it doesn't mean that the molecules, the gene products that these genes encode are not relevant. I mean, these enzymes and receptors and proteins that are produced by these genes have very central roles in human biology. But what our results indicate is that the DNA sequence variation that we find in these genes are not really affecting the level of these gene products to an extent that it would translate to differences in athlete or athletic performance. And the only way actually to solve this problem is to use an unbiased, hypothesis-free, genome-wide screening approach. The other problem we have, or we usually have, is how we define the athletic phenotype. We have a lot of studies in literature who state that they selected their subjects based on success in international competitions, for example, Olympics, World Championship tournaments, and so forth. However, at first glance, this seems like a logical approach. But then if you start thinking about it on more detail, there are so many factors affecting how you actually achieve or time your peak performance in terms of these big international events, that we most likely will miss a lot of good athletes, excellent athletes, elite athletes, if we simply look at the outcome of these competitions. For example, my favorite example is a Finnish cross-country skier, Juha Mieto, who lost gold medal in 15-kilometer cross-country event in 1980 Olympics by 1/100th of a second. So basically, he lost the race 15 kilometers by this much. And that was actually the last time they used hundreds of seconds to measure time in Olympics. So I don't think that loss would unqualify Mr. Mieto from being an elite athlete. So I think what we need to do is actually use some kind of combination of basic requirement for athletic success for our athletes and a physiological measure or a predictor of the athletic performance. On this regard, we also need to pay attention how we select our controls or non-athletes, because we know from human genomics and human genetics that depending on which part of the world individual comes from, that may have a drastic effect on what kind of DNA sequence variation we will find on a given group of individuals. So matching for the country of origin and ethnicity is very important in these the type of studies. So the genome-wide association approach that we hope will solve the problem of candidate gene approaches is based on really an unbiased, hypothesis-free study design. So that unlike in candidate gene studies, we don't try to understand the biology to select our target genes and DNA sequence variants. But in the GWAS approach, we let the genetic information tell us about the biology. And the way these studies have been applied or implemented in science basically has really changed over the last 10 years, because the first, or actually the whole era of genome-wide association studies was made possible by the advances in genotyping technology that occurred in the first part of 2000s. And in 2004, 2005, both Illumina and Affymetrix released their first so-called GWAS arrays. And these were array-based products at that time contained about 100,000 SNPs or single nucleotide polymorphisms on the chip. And that was truly amazing compared to the technologies we had before that. And then the field evolved very quickly, so that soon we had chips that contained 300,000, 500,000, 600,000 chips. And currently, the most dense chip we can use is 5 million SNPs in one array. So that with one assay, you can get information from over 5 million DNA sequence variants from one sample. The statistical testing with GWAS studies is very straightforward. Depending on the study design, you can use it with case control design, dichotomous traits, or you can measure your outcome variable on a continuous scale and use that as your trait of interest. The GWAS approach is also very useful in detecting fairly small effects of common sequence variants, especially if you compare those effect sizes to what we were able to detect with the previous genome-wide linkage analysis, so that the GWAS approach is much more detailed and powerful in that sense. However, the frequency of the individual alleles that we can utilize in current GWAS approach is about 5% and more, depending on the sample size we have. So the limitation then comes in terms of how many subjects we need to do this analysis, which, in other words, the target sample size is in thousands rather than hundreds. And then still we have the limitation that with these approaches, if we have really rare variants with fairly big effect sizes, we most likely will not be able to detect those using these approaches. So the GWAS approach should provide us a solution for the candidate gene problem. The other way to enhance our study designs is to use the physiological measures to aid our subject selection. And we know that high VO2 max is essential for endurance athletes to perform well in competition. So therefore, since we can measure VO2 max fairly easily in laboratory conditions, we should take advantage of that information when we select our athletes for genetic studies. Also, this by no means means that VO2 max is the only physiological trait that contributes to athletic performance, but it's definitely a very important one. And if we use high VO2 max as a criteria to select our subjects, we will most likely also concentrate our athlete group on those individuals who has the genetic predisposition to have really good aerobic capacity and therefore be suited for to perform well in endurance events. And also, finding these athletes with really high VO2 max will give us an access to those phenotype values that we don't usually find if we study just a regular population, and therefore it gives us other kind of information on VO2 max as health indicator. And we know also from studies, what Dr. Bouchard already reviewed, that both the sedentary state VO2 max as well as VO2 max training response are both heritable traits with heritability estimates around 50%. So what we wanted to do with the award we were granted was to take advantage of the genomic technology and our gene athlete cohort, where we used the VO2 max to select our athletes and really do the first genome-wide exploration of genetic variants that would be associated with elite endurance athlete status. And the cohort we used for this study is the gene athlete cohort that Dr. Sarasinski already described. And so this cohort consists of currently 315 male endurance athletes. And our requirement was that their VO2 max has to be at least 75 ml per kg per minute. So much more than my own VO2 max. We recruited these athletes from North America, USA, and Canada, from Germany and from Finland, and we selected a one-to-one ratio of male non-athlete controls from the same countries. And also we put a requirement for the controls that they must have a tested VO2 max less than 50 ml per kg per minute. For genetic screening, we used selected Illumina CardioMetaBot chip, which includes almost 200,000 SNPs. And this product is not a classical GWAS chip, but it is basically designed based on multiple GWAS studies already reported in the literature, and they selected, a group of scientists did a very rigorous selection process of including SNPs that have been shown in previous studies to be associated with cardiovascular and metabolic phenotypes. For example, blood lipid levels, blood pressure, cardiovascular disease, type 2 diabetes, and so forth. And then so basically this chip could be used either for further further confirmation of the discovery studies or to conduct completely new studies. And after we did our quality control system, we find that we actually had 145,000, not 145 as the slide says, but 145,000 SNPs that were polymorphic and passed the quality control filters that we used for the study. The reason why we lost that many SNPs due to to monomorphism is that the chip is designed to cover not only Caucasian populations but also African Americans, Asian populations, and other ethnicities, so that those many times those markers that were selected for example, African Americans are simply not polymorphic in Caucasians. So that's the reason why we lost almost 50,000 SNPs at that stage. And then the statistical approach we used is very simple allele frequency comparison between athletes and controls using chi-square test. So again, here are the basic characteristics of our athletes and controls, so that exactly the same height. Athletes were less heavy than controls, which is not surprising. The mean VO2 max in athletes 79, 40 in controls, and we had the equal representation of all three countries of origins among athletes and controls. All right, so here is the summary of our results in terms of Manhattan plot. And so what we have on the x-axis is individual chromosomes, and the p-value from the association test of individual SNPs is then showed on the y-axis. And each one of these dots represents individual SNP. So as you see, we have a few cases here where the level of association clearly separates from the background noise shown here at the bottom of the graph. And this table shows you the most significant SNPs we found with using our 315 athletes and 320 controls. And the most statistically significant association we found was with the group of SNPs located in chromosome 15. And this is a region that is described as a gene desert. In other words, there is no known genes in near proximity. But the reason why these markers were originally selected for MedAPA is because in Framingham Heart Study, these SNPs were very strongly associated with cardiac arrhythmias. So we picked up exactly the same SNPs with our gene athlete samples, and now we need to do further work to really understand if there is a physiological or biological link between the arrhythmia findings and our elite athlete status findings. And in terms of the effect size, the OR or odds ratio column indicates the strength of the association, so that all odds ratios that are above 1 basically means that the allele, the effect allele, is more common in athletes than in controls, and the odds ratios below 1 means that the effect allele is less frequent in athletes than in controls. And as you see, most of the effect alleles we identified are more common in athletes than in controls, and usually they increase the odds of being athlete by 60 to 150%. All right, so this is these are the results in terms of individual SNPs. But we know already from literature that it's very, very unlikely that the athlete status will be explained by one single SNP. Instead, we most likely will have so-called polygenic effect. In other words, multiple SNPs will contribute to the athletic status. So, in order to describe the joint effect or joint contribution of multiple SNP, we apply a method called genomic summary score. And basically what this means is that since each SNP has two alleles, and one inherited from mother, one inherited from the father, and so first thing we need to do is once we know which SNPs we want to use to generate the summary score, we first identify for each SNP which allele is associated with the trait that we are interested in. For example, in our case, more common in athletes. And then we simply calculate how many desirable alleles each subject has for the SNPs we use for the score calculation, and then we sum the number of positive alleles across all SNPs, and we finally end up with the score where theoretical range of values ranges from 0, in other words, having no beneficial or detrimental alleles, to value of 2 times the number of SNPs we use for the score generation. So using this approach, we selected 8 SNPs, 8 independent SNPs from the previous table, and calculated the scores in gene athlete cohort. And here our athletes are represented with black bars, and sedentary controls are shown with the gray or bluish gray bars. And as you can see, the distribution of these summary scores is clearly shifted towards the higher values of the summary score in our endurance athletes compared to sedentary controls. So the combined contribution of these 8 SNPs is clearly different between alleles or between athletes and controls. Then we would like to, when we work with continuous traits, we can easily describe the contribution of our genetic markers simply by telling how many % of the total variation our genetic markers explain in our population. So we can put a clear % number on those predictors. But when we deal with binary classifier or binary outcomes, like in a case control studies, athlete versus non-athlete, that kind of description is more challenging. However, we can use the sensitivity and specificity of a test to accomplish that purpose. So again, just to refresh your memory from Dr. Bouchard's presentation, with sensitivity we really mean the true positive rate and of the classifier, and specificity refers to the true negative rate. So how many how many positives we get right and how many negatives we get right. And graphically, we can present this with receiver operating characteristic curves, and we can use this approach to describe the overall performance of the classifier, and also to identify the optimal cutoffs of a continuous predictor trait. In other words, the value that we can use as a cutoff to predict the cases and controls that has the best sensitivity and best specificity for the trait. So we, if we just take a coin and start flipping the coin and predicting our outcome variable that way, we usually get about 50% accuracy. So about half of the time we are right and half of the time we are incorrect. So what we end up with is this area under the reference line that reflects that chance alone. And if we present the specificity and sensitivity on a graph like this, what we want to have is a predictor that will go as close to this top left corner as possible. That's where we maximize or get perfect sensitivity and perfect specificity, so perfect or predictor. And usually what we end up with is something like this, where we have a moderate predictor described with a red line and a fairly good predictor described with the blue line. And and everything, any improvement in the prediction that occurs above this reference line simply then reflects the performance of our predictor. So we applied this approach to our genomic score or genomic summary score data, and this is the outcome we found. So that if we look at it as terms of estimated area under the RUC curve, we get value 0.7. And basically it means that it adds, and now remember the chance alone would be 0.5. So we add about 20 units in the area under the curve with this predictor. All right, so our original study design required that our athletes must have VO2 max at least 75. But we wanted to also test that what happens if we apply a bit more stricter cutoff for VO2 max among athletes. And among our athletes, the median VO2 max is 78. So we decided to use that value as a new cutoff and to select endurance athletes with very high VO2 max. So this approach gave us 168 male athletes with really high VO2 max. And again, we used the same 320 sedentary controls that we used for the first set of analysis I showed you. And this is again the Manhattan plot of the results. And now if you happen to remember by heart the y-axis from the first graph, you see that here we have a bit more significant associations. And basically two things happen. We still see the same SNPs we already saw with the full cohort. For example, the chromosome 15 is still there, but we also add new loci that were not quite as readily detected with the full cohort. And the other feature we notice is that our statistical significance with this subgroup analysis is much higher than what we saw with the full cohort. Now these odds ratios are ranging or reaching 2 and even more than 3. All right, so if we follow exactly the same steps with the genomic score marker, again using 8 SNPs, the separation between athletes and controls is even greater than in the whole cohort. Again, the RUC curve, this time we are we have estimated area under the curve 0.77. Basically, we improve from 0.7 to 0.77 compared to the full cohort. All right, so that's basically the basic results as we see them in the full cohort and in high VO2 max sub-cohort. So the really interesting issue, first of all, is that although we didn't see anything that was clearly genome-wide significant after multiple testing correction, we have several SNPs that had really interesting trends for association. And these were with genes or within gene loci or chromosomal regions that have been previously reported in sedentary individuals or non-athlete individuals to associate with cardiac function or metabolic regulation. We also, and let's also remember everything we found in the full cohort and in sub-cohort of high VO2 max athletes were in loci that we would not necessarily have selected as a candidate gene loci. So that alone tells us that the genome-wide exploration actually worked in our favor. Then second clear issue that we notice is that our results clearly support that polygenic genetic model hypothesis for endurance athlete status. So we have several SNPs and genes with small to moderate effect sizes, but we didn't find that single major gene in our cohort. And most likely that single major gene probably doesn't exist. And the interesting thing is that if we really took advantage of our physiological athlete qualifier, in other words, the VO2 max measurements, so if we applied even stricter cutoff for the desirable VO2 max, we saw slightly different results. So that we detected some genes, for example, the adrenergic receptor gene, that we really didn't see come up as clearly in the full cohort. And also the effect allele frequencies among the 168 athletes with the really high VO2 max, those allele frequencies were fairly low in general population. When we are talking about frequencies between 5% and 10, 11%. And this raises the interesting question that do we really have a case, a truly polygenic case, where the genetic architecture underlying elite endurance athlete status is truly consisting of multiple rare to low frequency variants that we probably will need even more sophisticated technologies, next generation sequencing to truly capture among our athletes. And so we already now have pretty good leads on where to go next with our studies. But one thing we have learned with genetic studies over the last 10 years or so is that the replication of the discovery results is extremely important. So therefore, we have made initial contacts with several colleagues in Europe and in Japan to find athlete cohorts from different regions of the world to actually be able to test these markers that we have identified. We have runners from Asia, from Africa, from Japan, Spain, Australia, and we would love to have similar cohort of Arabic athletes because the only way we can create a universal genetic predictor, is if we have information from all around the world. So I will finish my talk by expressing our true gratitude to Prince Faisal Foundation for giving us the opportunity to do this exploration. And also I want to acknowledge my co-authors, Dr. Wolfart, just back from Sochi Olympics, Dr. Sarsinski, Dr. Perus from Quebec, Dr. Raurama from Finland, and of course Dr. Bouchard. All right, thank you.
كما يعني بانت خبرتهم في ضبط الجينات، فهم أيضا بانت خبرتنا جميعا في ضبط الوقت، واللي شيء مهم جدا بالنسبة لكم. الآن إذا فيه أي أسئلة ممكن تطرح على الخبراء هنا في هذا المجال، تفضل.
بسم الله الرحمن الرحيم. أستاذ دكتور عبد الرزاق عبد الجبار حمزة، أستاذ علم التدريب الرياضي وخبير بالانتقاء الموهوبين. نشكر الأخوة القائمين على جائزة الأمير فيصل بن فهد بن عبد العزيز على دعوتهم الكريمة وفسحهم المجال للقاء بفريق من العلماء والباحثين في المجال الرياضي. حقيقة هناك سؤالين، سؤال يتعلق بالمنهجية العلمية، وسؤال آخر يتعلق في البحوث اللي حول الجينات. السؤال الأول للبروفيسور بوشارد، أنه عينة البحث اللي يختارها سواء كانت عمدية أو عشوائية، لاحظت أن هناك عدم وجود تجانس أو تكافؤ في عينة البحث، لأنه نسبة كانت الانحراف المعياري كبير جدا 32.5. فعندما يكون الانحراف المعياري كبير جدا بالتأكيد هناك تشتت، وهذا ينعكس سلبا على النتائج اللي يتوصل إليها، راح تكون نتائج غير معنوية، وبالتالي لا يمكن تعميم هذه النتائج. السؤال الآخر للفريق بالكامل هو حول مسألة البحوث التي أجريت على الجينات. بالتأكيد هاي البحوث العلمية الحديثة فيها تساعد في عملية الاقتصاد في الجهد والوقت والمال في تشخيص رياضي النخبة. من المعروف سابقا أنه كان من خلال نوع الألياف العضلية إذا كانت بيضاء أو حمراء أو مختلطة، ممكن أن نوجه الرياضي إلى نوع النشاط الرياضي التخصصي الذي يناسب نوع هذه الألياف. الآن البحوث الحديثة اللي بدت مثل ما قالوا من 1968 من دورة مكسيكو سيتي، أنه بدت بحوث في مجال الجينات. وهل هذه الجينات، هذه الاختبارات، هذه البحوث على الجينات ممكن أن أحدد نوع القدرات البدنية للرياضي حتى أقدر أوجهه لنوع النشاط التخصصي اللي يناسب قدراته البدنية؟ وشكرا.
So the first question is there's two, and maybe the second one we can answer generally. Yeah, yes, briefly on that. No, you can speak here. Yeah, you can answer. You, Mark. No, you have one.
Okay. Well, thank you for the questions. Let me address the first one, I think, because it's quite fundamental. Subjects in the studies that are being done in all labs all over the world for exercise training or any other kind of exercise studies are not truly representative samples of the population per se, because you start with volunteers. You ask for participants and you take those who volunteer. So in that sense, they may slightly be different from the population. But if the sample size is large enough, which in the kinds of studies that we do, we have it is a fundamental principle in our laboratory that we have to have the proper statistical power, and therefore we recruit generally very large study sample size. In this case, I'm not too worried about the representativeness of the population, because we cover both genders generally. We cover quite often a number of ethnic groups and an age range. So we feel pretty confident that the generalizability of our observations is good. Now, the fact that we have a standard deviation which is high does not invalidate anything. It says that the distribution of the scores has some unique properties that we need to understand. And in the present case, it's easily understood. You referred to the response to fasting insulin in the heritage exercise training program. So we have a change of about 6 or 7 picomole, but a standard deviation 5 times larger than that. It means that we had a big skewness in the distribution. There were lots of people who were responding well, but you had a lot of people who were not, and you had some people who were getting a change in the wrong direction, an adverse response. So it was a peculiar curve, but it's still very representative of what's going on. That's what biology is in this case, and it reflects what we have always been proposing in our laboratory, and that is that human variability is one of the most important things to understand. The mean effect is not really that critical in science. It's the inter-individual differences that's informative in science. So, and then the second relates to how to use that information. You're right. The interest in finding inherited traits that have the ability to distinguish between those who are well endowed to succeed in sports at a very high level goes back about 5 decades. And it started with questionnaires, and then it started with blood groups and with the HLA system and with enzymes, polymorphic enzymes from skeletal muscle and through isoelectrofocusing, and we changed technologies about 6 times since we began looking into these questions. And today we do unbiased, hypothesis-free screen of the genome, and we let the chip fall where they fall. And what Dr. Rankinen has shown, and what I showed on the screen we did on Heritage, is that the chips are not falling where we were looking. We were looking where there were lights, right under the spotlight, because these were our candidate genes. We were looking there. These unbiased genomic screens are telling us that it's not there. It's elsewhere. It's in pathways that we had not considered before. But the question still remains, even if we have the right pathways, the right networks, the right systems biology, the right genes, and maybe the right genomic markers, are they powerful enough? Are they precise enough, sensitive enough, and specific enough to be used with children? And at the moment, it's no, they are not. You will do more harm using them now than just advising them to continue doing what they enjoy. because if you use them, you're going to be saying to some people, you don't have the talent, while in reality, they may have the talent. And you're going to tell others, no, you're not talented, and in practice, they may be talented. In other words, we don't have enough sensitivity and specificity. You saw the curve that Dr. Rankinen showed, the rock curve. It's still an improvement of only 27%. In the best case scenario, 27% improvement over randomness. What we need is, we want to go to 95%. If we're there, yes, we will be justified to use them because we will not do any harm. We will be basing our evaluation on the truth. But at the moment, it'll be too risky. If you have a tall guy, very tall guy, and he's skilled, you may be right in guiding him or her toward a sport requiring these qualities, like basketball, for example. But if you're relying on genomic markers and you to override any hints you may have about someone's morphology and interest for a sport, to guide him or her towards a sport where you think the genes are pointing into, that is premature. And you have many companies now offering genotyping services to help you find which sports is right for you. And it really borders on the ridiculous, because they use one marker, sometimes more, a dozen or so, and they will tell you based on your genotype that you will be successful as a rugby player. My gosh, makes absolutely zero sense. This has no power, this has no specificity, this has zero sensitivity. So the only way to resolve this, I think that this research line has merit. Eventually, we will be able to deliver, but it will take research. We're not there yet.
ممكن أن أضيف على ما قاله البروفيسور. عادة الأبحاث اللي تجرى الآن هي ما تزال في عملية التنبؤ، وكما قال ليس هناك نتائج دقيقة، لأن عمل الجين لا يعتمد فقط على تركيب الجين اللي العلم الآن بيبحث عنه كسنب، بل يعتمد أيضا على تفاعل هذا الجين مع البيئة. وممكن تسيطر على الجين، لكن لا يمكن أن تسيطر على البيئة ولا التمارين اللي تعطى. وهذا المنطلق أو هذه الفرضية أيضا تبنى، لأنهم هذه مراكز أيضا تعنى بأمراض السرطان وأمراض السكري، محاولة التنبؤ أو أقل حاجة محاولة هل هذا عنده القابلية للإصابة بهذا المرض. كما قال ولخص البروفيسور على أن النتائج تعتبر بداية، لكنها الدقة لم تصل إلى الدقة كما نتحدث عن الجانب الآخر، كما هو البصمة الوراثية لكثير من القضايا. فليست هناك دقة في لكنه البحث بادي، والظروف المؤثرة أكثر من الجين نفسه. والجين كما علمتم من الجينوم البشري أنه نظرية الجين واحد، جين مقابل وظيفة واحدة، نظرية خاطئة، بل وظيفة واحدة يؤديها الجسم تشترك فيها عدة جينات بكميات ونوعيات محددة. فحبيت أن أضيف كذا ونستمع إلى سؤال الأستاذ الحسن.
السلام عليكم. أنا سأنطلق من مشوار البروفيسور بوشار الذي يهتم في مجالين أساسيين، ألا وهما عنصر الصحة، ونعرف بأنه له مئات الأبحاث منشورة في هذا المجال، خاصة فيما يتعلق بالسمنة. والمجال الثاني رياضة النخبة. وأبدأ بهذا العنصر الأول أو الأخير. ففي ظل صعوبة تحديد الجينات المرتبطة بالأداء الرياضي، خاصة لدى رياضي النخبة، وفي ظل الاختلاف الكبير في المنهجية المتبعة في دراسة الجينات المرتبطة بالأداء الرياضي، هل هذا يعني أننا لازلنا بعيدين كل البعد على اعتماد الجانب الوراثي كعنصر في انتقاء والتنبؤ برياضي النخبة؟ هذا الجانب الأول. الجانب الثاني، يعني رأينا في الفترة الأخيرة اهتمام كبير بالصحة، وخاصة الجانب الوراثي، حيث ظهرت بعض المؤسسات التي تدعي بأنها بإمكانها تحليل الجينات المرتبطة بالصحة، وخاصة الجينات المرتبطة بزيادة أو نقص الوزن، وأنهم بإمكانهم تحديد هذه الجينات ووصفة ربما قد تكون سحرية لخفض الوزن. ونعرف أن الكثير من الناس تعاني من هذه المشكلة حاليا. فما هو رأيكم بروفيسور بوشار في هذه الأشياء الموجودة حاليا؟ وشكرا.
Okay, I was not sure. So the first one, I will not throw the baby with the bathwater. I think there is useful genetic information that's out there, but it all depends how you're going to use it. Someone who is involved in channeling children and adolescents towards in the sports pyramid could use some information on genes, but not as the single factor to support the decision to orientate someone in the direction of sport X as opposed to sports Y. I think it's all a matter of using what we have intelligently until we have better. But it will never, quite likely, will never be an all or none situation. You use everything you know about that person, and the genetic information is something else that comes to enlighten, the particular case of an individual, and support to provide you additional support for the decisions that are about to be taken. I think we are in that situation, and we're likely to be for a while in that situation. The genetic data are going to become progressively stronger, and we should recognize that. But by itself, I don't think they should be the only tools that we ought to rely on in order to take these career sports career orientation decisions. In the case of health, we are pretty much in the same situation, except that we have a lot more research. You know, it's very difficult to get the funding at the level of for example, the National Institutes of Health or the European Community to do sports-oriented research, particularly performance-oriented research. You do better if you study exercise and health. Then they are interested. And here, let's take the example of diabetes or obesity. We do have hundreds of genes that have been shown to be associated, hundreds of SNPs that have been shown to be associated with the risk of obesity or the risk of type 2 diabetes. But yet, and they have been done in very large cohort. For example, the latest study which has looked into the SNPs that were predictive of the risk of obesity, encompasses about more than 300,000 subjects. So here we don't have a problem with the statistical power. It's not an issue. And out of that, there are about 100 SNPs that have been shown to be associated with an increased risk of obesity. If you have a lot of risk alleles at these 100 loci, you're very much at risk. If you have very few, you have a lower BMI and less at risk of becoming obese. But even with 100 loci confirmed, statistically well established, we account for about 5% only of the variance in body weight among people. So yes, those are firm, those are solid now. We know for sure they are real, but it's only 5%, which means that we were maybe a bit optimistic before. We thought that with these technologies, we could easily grasp the biological, the genetic basis of the predisposition to obesity or type 2 diabetes, for example. We thought we could do it. But it turns out that the underlying biology is more complex. It's not only single SNPs. It's more regulatory element in the genome that may be rare. They are not captured at the present time by the tools that we use. So we need to, every time in genetics, every time we uncover something, at the same time we attain a new frontier, a new limit, and it takes a new technology to allow us to explore that dark matter that we failed to perceive. We pushed the boundaries a bit more, but then we realized we need a new technology to explore that new frontier, and that's where we stand. We recognize now that the risk of these diseases is only to a very limited extent in the common variants that we exhibit in our DNA. It's in the common, because the common ones are the ones that we are testing with the tools that we have nowadays. So the next frontier will probably be in the rare variants rather than in the common. And after the rare variants, there will be something else until we have a full grasp of the underlying biology. That's where we stand.
طبعا ننهي بأنه نقول كلما ازددت علما بجهلي، وأن كلما زادت اكتشافات كلما كما ذكر البروفيسور وجد على أنه في هذه البداية، وهذه تعتبر على أنها يعني الجينات ليس بديل ولكنها مكملة للمعايير الأخرى اللي تختار أو تطبق لاختيار رياضي النخبة. وأنا حد يشير لي بالسؤال وحد يشير لي بانتهاء الوقت، فنرجو المعذرة كذا لأنه محددين نحن في الوقت، وإذا في أي شيء موجودين يعني في وتجد الجواب يمكن واحد يشرب شاهي أكثر من هو جالس على الستيج، فالمعذرة. أشكر ضيوفنا ومحدثينا، وأشكر حضوركم وصبركم على الإنصات لهذه الأمور اللي احنا بعض الأحيان نجدها معقدة بالنسبة لنا لفهم، والأساتذة موجودين في هذا الوقت البريك إذا في أي استفسار يسعدهم ويسعدنا جميعا أن نجيب عليه. شكرا. خلاص لا.
شكرا للدكتور محسن العامري والمتحدثين الكرام، دكتور كلود بوشارد، الدكتور مارك سارزينسكي، الدكتور تيومو رانكين. شكرا لكم سيداتي وسادتي. الآن استراحة لمدة 15 دقيقة إن شاء الله، لنعود بعدها ونستكمل الجلسات الشيقة اللي تتحدث عن العوامل النفسية والميكانيكا الحيوية لدى رياضيي النخبة. رجاء 15 دقيقة مش أكثر حتى نحافظ على وقتنا. شكرا جزيلا. أهلا ومرحبا بكم في الجلسة الثانية من الندوة. الجلسة الثانية تنقسم إلى قسمين، والقسم الأول سيكون بعنوان دور العوامل النفسية لدى رياضيي النخبة. يدير الجلسة الأستاذ الدكتور الحسن النبلي، عضو مجلس الأمناء وعضو اللجنة العلمية في الجائزة، ويستضيف فيها الأستاذ الدكتور تيري أورليك من جامعة أوتاوا في كندا، والأستاذ الدكتور عبد الرحيم بريع من الجمعية المغربية لعلم النفس. فليتفضل الدكتور النبلي وضيوف الكرام.
السلام عليكم. يشرفني أن أترأس هذه الجلسة الثانية في محور ثاني من محاور الجائزة، ألا وهو محور علم النفس الرياضي أو دور علم النفس الرياضي في الارتقاء بالأداء أو الإنجاز الرياضي. ويشرفني أن أكون بين اثنين من كبار علماء النفس في العالم، سواء العالم العربي وهو الدكتور بريع عبد الرحيم، وأحد رواد علم النفس الرياضي في العالم الدكتور تيري أورليك. ونبدأ بالمحاضرة الأولى ويقدمها البروفيسور الدكتور تيري أورليك، الذي هو أستاذ علم النفس الرياضي بجامعة أوتاوا، والذي أتشرف أني عرفته من الثمانينات لما كنت طالب بكندا، وتتبعنا مساره الطويل في الإعداد الذهني للرياضيين وأبحاثه الكثيرة في هذا المجال، التي أدت إلى اقتراحه لنموذج حول التميز الرياضي، خاصة الجوانب المتعلقة بالجوانب الذهنية أو العوامل النفسية في الأداء. وأصبح معروف كثير بما يعرف الآن بعجلة التميز The Wheel of Excellence. أدعو الدكتور ليقدم لنا صورة عن هذه العناصر الكثيرة، وخاصة ما توصل إليه الآن أن في هذه العناصر الكثيرة فيها عنصر أساسي ويعتبر قاطرة هذه العناصر، ألا وهو الكونسنتريشن. فليتفضل الدكتور ليقدم. Please. Thank you. Yeah.
Hello. Okay, can you hear me back there in that section? Good. So I'm going to share a few stories with you of recent people I've been working with. You know, I've worked with many teams, many Olympic teams, many professional teams, surgeons, astronauts, pilots, you know, all kinds of different performance areas. But I'll be sticking mainly to the sport area, although a lot of the same concepts apply to whatever you want to be great at or be good at. So this recent experience, one of our rowers, who was an Olympic gold medalist in the men's 4 rowing, and his name is Adam Creek, and he and 3 other rowers decided they were going to attempt to row unassisted across the Atlantic Ocean. So they were starting in South Africa, and they were going to row to the southern United States, and totally by themselves. No support boats, no one in there to help them if something goes wrong. So he called me and asked me if I would be willing to to help them prepare for that voyage. So I said, yeah, sure, I'd be happy to do that. And so they did go to South Africa, and I spoke with them when they were in South Africa about some of the issues that I was a bit concerned about. Like the boat they're in it's probably about from here to the end over here to maybe about here, and it's about this wide. So it's going to take about 3.5 months to row across the ocean. No one has ever rowed that way unassisted. Some people have gone up further where it's easy, it's closer, but no one's ever gone that route, and that's why they wanted to do it. So one thing I was concerned about is, let's say even with your family that there's four people and you can never get away from them. You're in this enclosed area, you can never leave them. You can't go for a walk, you can't go outside, you're always in close proximity, and plus there's a lot of challenges, and they're tired and they're hungry, and they had to bring all their food with them, dried food and planning. And so one thing I talked about was highlights. Like I wanted them to record every day, to record their highlights. What were the highlights of the day for them? To be focused on the positives, because not every day is a highlight when you're out there in the middle of the ocean. And so they did do that. Like they had a little recorder, and they'd go every day and they'd talk about what their highlight was for today, the day, and also lowlights, things that were maybe causing a problem that maybe they could find a way to solve, if there were some conflicts. And these are big, strong guys, so I was concerned if somebody really lost their temper or got mad, and they could really hurt someone or throw them overboard or whatever. So I was a bit concerned. So we focused on and they called that roses and thorns. Like the rose, the red rose was like the happy things in the day, and the thorns were those things on the stem that can stick into you So we talked about positive communication, supporting each other through the journey, because there's going to be challenges on the journey. And I was able to be in contact with them sometimes. I was on the phone with them one day for about an hour, talked to each one. And their start, the currents were against them. So like we're rowing this way to go, that way, but the current is pushing them back towards the shore. So they had days where they were rowing. Their general plan was 2 guys row at the same time, so you 2 are rowing now. You're going to row for 3 hours, and then you swap, and then the 2 of us row for 3 hours, and 3 hours, and 3 hours. 24 hours a day they're rowing. And so you're not sleeping that well, the boat is tossing. So like about 5 days in, they're going backwards because of the currents and the winds. They're pushing against them. So it was like trying to keep going and keep people positive during this whole experience. So I got an email from Adam Creek at one point where he said, I don't even know what we're doing out here. The weather's bad, and we're getting pushed back by the currents, and he's a very positive guy. So I was a bit concerned when he was not positive. He's one of the most positive people that I've ever met. So I ended up, after that night, I wrote this, what I called the 10 Commandments of Focus. And I sent that to him because they had a computer on board, and it was solar powered. So as long as they could see the sun, they could get messages, and I can get it. So I'm going to give you what these 10 Commandments of Focus were. And, in the end, when I got feedback from them at the end of 3.5 months, this really helped them stay focused on things that could help them move forward in their mission. So the first was to focus on what's within your control. This is all stuff I've used with other performers and other athletes, but in this situation, there was some urgency for them to be staying positive, because there were a lot of things that weren't positive. One day there was a storm. They had 2 spare oars, and it broke both of those oars. They had no spare oars left. If they lost another one, they wouldn't be able to continue. Things like that. So this is a big one. You don't control the currents, you don't control the wind, you don't control the weather. You just focus on what you control. And what is that? That's pulling a stroke, pulling a stroke, pulling a stroke, supporting each other. So that was one. Let go of what you don't control. Like, don't waste all your energy on things you don't control, because you don't control them. So it's not worth the energy. You can't waste the energy on that. Focus on the next step, next stroke, next move, because that's within your control. That will keep you moving in the direction you want to go. I mean, this is not only for them in this boat. This is for anybody in your life or your performance or whatever it is that you want to do. And that was, they can always pull one stroke, no matter what's happening. You can pull it one time, and then you pull it another time, and you pull it another time. And that's what you got to focus on, because that's what you control. And looking for opportunities, always looking for opportunities was the fourth one. This other one was, being kind to yourself and others, supporting each other, especially through the challenges of the journey, where you're supporting one another to help get to where you want to go. Because if you're being negative or arguing or complaining, it doesn't help anybody do anything. It's not going to help you complete that mission. So he stuck that up on a place in the boat where everybody could see it. And on my call with him, I said, Did you put the 10 Commandments of focus up there? And he had them all in his head. He said, number 1 was this, number 2, number 3, number 4. Not that they have a lot of other things to do out there other than, pulling those strokes. Yeah, so that's the boat. This was the thing on the highlights. Every day, look for the positives. Continue to look for the positives. Look at how far you've come. They finally started to move and get moving in the direction that they wanted to go. Looking at how far they've already come. Because if you're just looking at how far you have to go, it could be a little bit deflating in terms of how are we ever going to finish this thing. Focus on reasons to believe that you can complete your mission successfully. Like, why can we do this, rather than why we can't. And focusing on how you will successfully complete your mission. So in the end, that's the boat as well. Finally, the currents started to change a bit, and finally they started moving in the direction they wanted to go. And a lot of the highlights were nature things. They had hundreds of dolphins following the boat. They had whales out there. They had flying fish that just came into the boat that gave them extra food. There were a lot of good things that were happening, and then they're getting closer and closer. Every stroke is this much closer to getting across the ocean. They did have they were moving along well. They were out there then about almost 3.5 months, and they were 4 days away from arriving in the southern United States, in Miami, and a huge storm came up, and there were waves that were like as high as the top of this room, and their boat completely tipped over, so it was upside down, and it's like these rogue waves. So they got thrown out of the boat. Adam was in, if we go back to that shot, he was in the front underneath, like sleeping at the time. So when the boat went over, when he woke up, like the place was filled with water. The boat is completely upside down, and he's trying to find air. He thought he was going to die because he was underwater, like didn't know it was night, it was dark, but he managed to get a few breaths of air up at the top here and found his way out, and so they didn't quite complete the mission, but they were 4 days off, and they all survived, and they have these distress calls that you can on their life jackets. So when the boat went over, one of them pushed that signal, and then any boat in the area, the law of the sea is they have to come and try to help people who are in trouble. So there was a huge freighter that was coming across the ocean that got the signal, and then they managed to get over there and get on that big freighter. So they were, they did get to shore successfully, but they didn't quite do it those last 4 days on their own. So yeah, they arrived and they were fine. So I have come up from my working with so many great performers, I've come up with these different wheels. The first wheel I did, this all comes from people's experience. What allowed these people in so many different domains to excel. And commitment is probably the most important first one. Like you are not going to be great at something or reach whatever your potential is. You're not even going to get close to it unless you have a huge commitment to get to where you want to go, to do this mission. And mental readiness is another one, like every day being mentally ready for what you want to do today, getting ready on the way in. The whole thing of having positive images of what you want to do, what you're capable of doing, how you're going to do it, like running through that stuff mentally before you get into the real situation. Developing a confidence in yourself that you can do the mission. Distraction control is a huge one because that's usually what throws you out of your game or out of your best performance. And grabbing the lessons from every experience. If it was great, if it was a great day, a great performance, a great whatever it is you're doing. So like what helped that happen? What did you focus on to make that happen? And if it didn't go well, what was going on then? And just then doing some kind of a debrief, whether it's in a performance setting or any other setting, reflecting and grabbing the lessons learned that can help you be better tomorrow or the next day or the next day. And that's what Olympic gold medalists or world champions or people who are the best in the world at what they do, they have all those things working for them. And the center is, a positive focus, a connected focus. Be 100% where you are. Don't be here if you're not here. If you're not here mentally, you might as well be somewhere else because you're wasting time. The second wheel is the wheel of highlights. So here we're trying to enjoy as much of your life as possible. To excel at living, not only excel in the performance domain, but some people are excelling in performance domains, but they really don't have a life. that's that exciting outside of the performance domain. So these are areas of highlights. I've done a lot of studies with athletes and students about what are the things that make your life worth living. You know, like what are the things that lift you, make you feel happy or joyful, or so this is from thousands of people keeping journals. These are the things that seem to be the major things that keep people engaged and happily engaged in their life. Human interaction is one, nature is another, relaxation is another, accomplishments, positive sensations. That could be a lot of different things. Personal discoveries, play, physical activity, sport. And if you can be positive and fully connected in these different domains, then you will experience more highlights every day of your life. And if you don't, then you won't. You know, if you look at people who are unhappy, they are not embracing a lot of these things. You know, it's not that they're not, those opportunities are not there, they're just not embracing them. You know, you walk out the door and you don't even see the beauty of being out that door. You know, or you're not fully connected. You're with somebody, but you're not really there. You're there physically, but you're not really there. You're not really connecting with them. So that's something that if you keep a highlight journal at the end of the day, writing down the things that were the things that made you feel best or gave you good energy in the day, you start to see what are the things that really lift you and what are other things that could lift you. And this kind of thing we're doing with little kids too. A lot of what I'm doing now starts really young, so that people can have these skills that they can carry with them for their life. And the third wheel is this wheel of focus. And so this is also something I'm doing with children, and like elementary school children and people at all ages, but starting with children. Like being focused on listening, you know, listening to learn. Don't just, you know, you're sitting there, I mean, even now in here, okay, you're here, but are you really here? You know, are you really listening? Are you going to remember anything, or are you not going to remember anything? I mean, you have a choice. You can be here and not be here, or you can be here and really be here. And the same with kids in classes or university students. They're in the class, you know, and they're there physically, but they may not be there mentally. So you're just wasting your time. So why waste your time? If you're not going to be here, don't be here. Basically what it is. So it's on listening and seeing things. When you see them, really connect with them. Feelings, connecting with feelings. Playing and performing, where you're really positive and you're really connected. Reading, you know, sometimes you read a page, you read a chapter and say, okay, what was in it? I don't know, you know, I don't even know what was in that chapter. So, like, what's the point of reading it then? I mean, you read the page to remember something, not just to, otherwise it's not even worth it. In learning context also, like being fully there, embracing the opportunity, in relationships, or whatever you're doing, if you're really something that you love to do or you enjoy to do, that you really connect to doing it. Because this is about living your life more fully. And it's also about performing closer to your potential more consistently. Because sometimes you're really good and then you're no good and you're here and you're there, and it's not that your physical or technical skills are changing usually, it's just that your focus is not where it can help you or free you to be your best or perform your best or think your best or write your best or whatever it is you're doing. And here again, the reflecting on, that you reflect on these things in order to improve or embrace more parts of your life or more of your life in a full and complete way. This isn't this another Olympic shot. Yeah, so these are things that from my work and research in different areas with real people doing things at a high level, positive focusing skills are really essential to be the best you can be. You have to focus in positive ways. You got to be fully connected, and these are things where we would have a plan. Okay, so what are you going to focus on? How are you going to make sure you're focusing in positive ways? What are you going to be thinking about? What are you going to focus on to be positive about this situation? What you can do within it? Fully connected focusing skills, like where you are there from start to finish. You're totally here. You're totally here. You know, you're in the moment. You're not here and not, disappearing somewhere else. Distraction control skills are probably one of the biggest ones because there are a lot of distractions in performance situations, or like in their rowing situation of going across that ocean. There's a lot of distractions, and sometimes they're internal things getting in your way, and sometimes they're outside things. So we have distraction control plans. Okay, so what if this happens? What are you going to do? How are you going to get back on track? Well, I'm just going to do that, take a breath, focus on this. So there's a lot of planning to make sure that these are distractions that you might expect in this context or in an Olympic context or whatever it is that you do. So you know these things are going to happen. So how are you going to make sure that you don't let them interfere with your performance? Refocusing skills are big, really big, and especially when in events that count. So shifting from negative to positive. Also, if you don't plan that, often it doesn't happen. Or it takes too long. You're spending a half hour being negative, and before you shift to a positive. So refocusing, negative to positive, refocusing, disconnected to connected. That will lead to consistency of performance close to your potential. The whole area of relaxation and stress control skills is really important. It's not all about, in a performance context, especially at a high level, it's not all about more is better. Sometimes less with quality is better, and you need recovery time. You need recovery time. Your body, you need it mentally, you need it physically. And the ongoing learning, even people at the highest levels, taking the time to do what I would call a debrief, to reflect on the experience or reflect on the performance about, okay, so I'm happy with this is what I did well, but this is what I can improve. This is what I was focused on when it was going best. So this is another thing. Sometimes people have doubts. Even people, you'd be surprised, even people at very high levels, they may be wondering, I don't know if I can do it again. They've been, among the best in the world or the best in the world on some days, and they're still thinking, I don't know, I'm not sure I can do this. Or, doubts will come in, and you probably wouldn't expect that looking at these people from the outside, but it's quite common that you have doubts. So that's where I'm saying, okay, focus on reasons to believe in yourself. I get them to write down, tell me why you can achieve your goal. Remember, there was one alpine skier. I was in the airport on the way going to Europe for world championships, and we're sitting in the airport, and she's saying, I don't think I'm going to do that well, and normally I just let people talk, but I said, I don't want to hear why you can't do well. and I gave her a piece of paper and I said, okay, write down, tell me why you can have a great performance. And there were a lot of good reasons. You know, she had some of the best races of her year, of her life that year. She was prepared for this. She had done that. She was rested. You know, there's a lot of good things. And so she would write all those things down, why I can. And then the other sheet was, and how I will. So these are my reasons to believe, real reasons, and this is what I will focus on to make this happen. And she ended up having an awesome performance in that. But these doubts, if you're thinking to yourself, I can't do this, well, you know, it's probably not going to help your performance. And if you think about why you can't, if you think about why you can and how you will, all of a sudden how you will always comes back to focus. This is what I have to focus on to make this happen. I mean, acting on your positive intentions every day is important, because otherwise you're not going to get there. Finding simple joys, embracing them every day, acting on lessons learned. And I'm a big one for highlight journals too, where you actually write down or take pictures of things good things, you know, feel good things, because that's going to give you energy that can help you be where you want to go. The other thing, I mean, some of the things you guys were talking about this morning, you know, I was thinking about this choice thing. You know, I think a lot of performing at a high level or achieving things at a high level, whatever it happens to be, it's a choice. It's a choice that you make. You know, you can choose to be a certain way, you can choose, and if you don't choose to do something every day to get to that goal, it's not going to happen. You can't just think about it, you know. You have to actually make it a priority in your life. A lot of people say, oh yeah, I want to be great at this, I want to do that, I want to be better at this, but they don't actually do anything every day to take a step closer to where they want to go. And if you don't do that, you are never going to get there, not in high performance context. Because if you're not doing it, somebody else is. You know, so you really have to make the choice. Is this the priority in my life? Is this, you know, what's your dream? Thinking about the dream goals. Maybe you're not quite there yet, but this is what I really want to do. And here's what I'm going to do today and tomorrow and the next day to get that much closer to that goal. And I have them write this stuff down, put it in writing so you can see it. Yeah. True. Okay. Yeah. So I'm saying. I mean, I believe now it's a choice. Personal excellence, performance excellence. You know, embracing your life. It's a choice. You either make the choice or you don't make the choice. If you don't make the choice, it's not going to happen. Even in terms of being more joyful in your life or doing the things you really want to do. Oh yeah, so I'll end on this little one. Can you see that one? It's a little heart. That is snow for people who've never seen snow from this part of the world. That's a lake that's frozen. That's where I live, on this lake. And that was one of my daughters when she was a bit younger. But I was talking to her about following a path with heart. You know, like you got to follow a path with heart. She said, "Well, what's a path with heart?" I said, "Well, I mean, it's kind of doing all the things you want to do and embrace them." And so we went out on the lake that day, and she had that hockey stick. And we were walking across the lake, and she started drawing little hearts on the snow. So I came back and said, "What are you doing there?" She said, "I'm following a path with hearts." She had hearts all the way across the lake. So anyway, that was that's kind of a perspective embracing your life and teaching people at early ages to embrace their lives, embrace the simple things, the simple joys, the simple opportunities, and just try to keep moving in that direction. Okay, good. We're done. 30? Okay. You're on, right?
Thank you very much, Terry Orlick.
You're welcome.
أشكرك جزيل الشكر تيري أورليك على احترامك للوقت المخصص لكم، وأود الإشارة أن ما ينطبق في علم النفس الرياضي من تقدم في العلوم أو في المنهجيات هو نفس التطور الذي تكلم عنه قبل قليل البروفيسور بوشار، أن نتائج الأبحاث تأخذنا إلى اعتقاد بأن وصلنا إلى مفهوم معين للأداء الرياضي، هذه النتائج تكون في الواقع هي الأساس لأبحاث أخرى تجعلنا نعتقد بأنه قد توصلنا مرة ثانية إلى نتائج تمكننا من تفسير الأداء الرياضي، لكن نكتشف مرة أخرى بأن هذا الأمر ليس إلا منطلق جديد للأبحاث في هذا المجال. فمن خلال تتبعنا لمسار الدكتور البروفيسور تيري أورليك نعلم بأنه في الثمانينات قد اقترح نظرية للتميز الإنساني، ثم اتضح بأنه ليست للتميز في جميع بالمطلق، بل هو تميز في عناصر معينة، واقترح آنذاك عجلة تميز من 7 عناصر كانت نواتها عنصرين. في التسعينات ظهرت ما يسمى ب 3C، يعني 3 عناصر كلها تبدأ بكلمة C، واعتمدها الدكتور في 2000 على أساس أنه فعلا النواة تتكون من 3 عناصر و4 كإشعاعات، لكن في تقدم للعلم اتضح له لا، بأنه والله في عنصر أساسي هو عنصر التركيز. الآن نكتشف بأن الدكتور يقترح أشياء أخرى، عجلات أخرى وليس عجلة واحدة، عجلة للإشعاع ثم عجلة للتركيز، وهذه قد تكون منطلق جديد مرة أخرى لدراسات في هذا المجال. هذا فيما يتعلق بمداخلة البروفيسور تيري أورليك. الآن ماذا عن العالم العربي؟ هل دراسات في مجال علم النفس الرياضي وصلت إلى هذا المستوى؟ أو هل الرياضيين العرب لهم نفس المنظور حول العناصر المتعلقة بالتميز؟ هذا ما سيتطرق له الأستاذ الدكتور بريع عبد الرحيم في بحثه المشترك مع الدكتور تيري أورليك، وهو الفائز أصلا، هذا البحث الفائز بالجائزة. البروفيسور بريع عبد الرحيم أستاذ علم النفس الرياضي بالمدرسة العليا للأساتذة التابعة لجامعة الحسن الثاني بالدار البيضاء بالمغرب، نائب رئيس الجمعية العالمية لعلم النفس الرياضي، رئيس الجمعية العربية لعلم النفس الرياضي، رئيس الجمعية المغربية لعلم النفس الرياضي، وله الكثير من الإنجازات في هذا العالم. وربما من الأشياء التي توجت مساره المهني فوزه مع البروفيسور تيري أورليك بهذه الجائزة في بحوث تطوير الرياضة العربية، فليتفضل مشكورا لإلقاء محاضرته.
بسم الله الرحمن الرحيم. السلام عليكم. أود في البداية أشكر الأخ والصديق الدكتور حسن النبلي على هذه المقدمة الطيبة، وأود كذلك أن أن أستسمح للحضور الكريم وأستأذن من الحضور الكريم بتقديم هذا العرض باللغة الإنجليزية، لأنه العرض البحث قد قام تحضيره باللغة الإنجليزية، فلذلك نتمنى إن شاء الله بترجمة هذا البحث في الأيام القادمة إن شاء الله. البحث الذي the presentation I'm pleased to present you today is about coaching the focusing skills required to empower Arab athletes to excel at the highest level. This presentation was conducted by Terry Orlick from Canada, from University of Ottawa, and عبد الرحيم باريا من المغرب من الجمعية المغربية لعلم النفس الرياضي. I would like first of all to present an outline of the presentation of this study, which is, in my point of view, has two sections. The first section relates to the reality of sports in the Arab countries. Not just for sports, but sport research, sport psychology, applied sports psychology, publication in the Arab countries, and all the studies that have been done and were related to the achieving excellence in sport. And I will also talk about some research that have been made about the factors associated with athlete success. And I will talk about our study, the practical study. Regarding the Arab elite sport, I would like to present some facts. And I begin with the first one, which is regarding the level of high-level sport in Arab countries. And following up Arab sport, we regret that the major international sporting accomplishments of the Arab countries are so modest. And if we compare our level as Arab countries, we can note that we are to some extent behind international sport achievement, taking into account our population, our potential, our human resources, and also our material resources. From Amsterdam, 1928, to London, 12, we can note that this level is still in its place, and we can note that all Arab countries doesn't exceed 105 medals from 1928. The 12 medals won in London for the last Olympics confirmed once again that all the Arab athletes have talent. There is a relatively low level of Arab athletes in many sports reaching the highest level of performance. Here is a table which indicates all the medals won by the Arab countries from 1928. In terms of sports psychology research, and it's a great point that we have to know it, sport research, unfortunately, doesn't seem to follow the evolution of sport performance in the Arab countries. Most of the studies we have noted or conducted are descriptive and limited till now, unfortunately, to theoretical psychological concepts. And the major topics that were investigated in those research are related to personality, motivation, anxiety, attribution, and some of them are applied that have investigate some observation of athletes' behavior. These studies are not applied or not original or innovative. They generally extend earlier findings and are predominantly influenced by the work of Western sports psychology research, whose research is not targeted to improving athlete performance in our real world. In terms of applied sports psychology, which are now the leading research in the world in sports psych, we note a low productivity rate. As Abu Shadi et al., Abu Shadi is the researcher that have won the 7th edition of the prize of the Prince Faisal. He stated that because the level of professional involvement with elite athletes is so modest. So in order to develop relevant applied sports psychology research in Arab countries, it is now crucial to make a shift from this traditional research to an in-depth and applied ideographic research paradigm that can found and uncovers the real factors that lead in consistent high-level performance. In terms of Arab sports psychology publication, currently there is no referred journal in sports psychology in Arab countries. But we can note that a number of studies have been published in proceedings and conferences or seminars in sports science in the Arab countries. There are a lot of, we have noted that many research are done, but majority of them are done in Arabic language, and we cannot find them in referred journal. Egypt, Jordan have an advanced university system in physical education and sport, including sports psychology. So they produce a lot of scientific literature in sports psychology and in sports science. Muhammad Alawi, just an example, who is the father of sports psychology in the Arab world, and some Egyptian pioneers have produced the greater part of book in this field. The evolution of the Arab sports psychology is, we can say that it is growing, but it is growing in a slow and modest rate. Professionals who provide practical and relevant intervention services to athlete and coach unfortunately remains rare. In last Olympics in London, I've been there and I have some meeting with some athletes and coaches from the Arab countries, and no country have a mental coach or mental trainer. This is partly due to a lack of practical applied educational opportunity available to Arab sports psychologists, as well as a lack of knowledge and notably the skepticism of some decision makers in charge of sport in our countries. About what? About the potential huge benefits of mental training and focused training for consistent high levels performance of excellence. High performance or excellence in sport is not an easy task. Achieving excellence in sport as well in other domains, it is a complex inquiry that requires the accurate combination of human talent and also an environment which can contribute to this achievement. In today's high-level sport performance, being physically or technically gifted, as what Dr. Bouchard explained this morning, is not sufficient to succeed at the highest level. An athlete's success requires high level of motor and mental skills and support from several sport-related sciences such as sport medicine, sport physiology, sport psychology, and so on. From the point of view of expertise development in sport, research concludes that international standards players in almost all sports took between 10 and 13 years to develop fully to international level. That's what we call the 10 hours of deliberate practice to become elite. The attainment of the high level of performance, as I said, may last a number of years, during which the athlete must focus on skill improvement in a well-developed, supervised training program. And what's about this supervised training program, which is lacking in the Arab countries? From the study that was conducted by Abu Shadi and his collaborators, this lack of a rigorous and comprehensive way of preparing athletes, or this mental training program, this lacking of those mental training program was among the constraints that prevent Arab athletes from achieving the highest level of performance. And what is a mental training program? A mental training program provides techniques and strategies that can teach to enhance and to optimize performance. These skills can be learned and can be improved and perfected, but through a consistent training. Psychological skill training, athletes and coaches worldwide are now more aware of the benefit of these mental training programs. This kind of mental skills training program continue, unfortunately, to be lacking in the general preparation of our athletes. It is most often in crisis situation and when facing defeat that in Arab countries we become to call a mental trainer, but he couldn't do anything at this moment. Compared to Western countries who are now accompanied by a mental coach to various international sport events because they recognize it as an advantage or a necessity. Over 1/3 of the American athletes who were at the last Olympic benefit from a mental training for the last Olympics. Few Arab athletes engaged in elite sport have ongoing access to a mental coach during training, and even fewer are accompanied by a sports psychologist at major games. In London, all the countries, Arab countries who participate in London and the village of the Olympic, no team or athlete was accompanied by a sports psychologist or by a mental training or has followed a mental training program. At present, a broad range of people from different backgrounds, unfortunately, NLP practitioner, clinical psychologist, with no graduate training or certification in mental training, become to be involved in this discipline. In the USA, for example, the Division of Exercise and Sport Psychology of the American Psychological Association state that to be a mental coach, you have to require some conditions. In terms of the mental factor associated with athlete success, we want now to see what are the research that was involved in this aspect. Research in sports psychology has accelerated over the past 50 years due to the growing awareness that winning edge in sport is not exclusively physical superiority or talent, but rather a mixture of physiological, technical, and mental factor. And now you see that performance is a systemic approach. It's not just one element, physical or technical, that can determine the performance. The identification of mental factors associated with high-level achievement has been one of the major topics of interest in Western applied sports psychology. Investigation dedicated to identify the mental component associated with this level of athletic success in Arab countries seems to be rare. Only a limited number of published articles report empirical data on psychological scales, and you can say that Al-Saqa and Nabli and Baria have done some investigation to determine the component associated with high-level athletes. For example, the result obtained by Al-Saqa revealed that indifference, concentration, motivation, psychological preparation, and control were the most important psychological skills for successful in sport excellence in the Arabia Saudi. But the mental links to excellence, the study that was conducted by Orlick and Partington in 1988, has been the forefront of qualitative studies that was designed to determine this component. The study have investigated 235 Canadian elite who participate in the 1944 in Sarajevo. Based on this result of the study with the Olympic athlete as well as individuals engaged in sport, and Orlick have formulated his first conceptual model of human excellence called the Wheel of Excellence that you have seen, with the core of this wheel is belief and commitment, and around it says 5 components with positive image, mental readiness, full focus, distraction control, constructive evaluation, and so on. This wheel have been changed through the experience and the studies that Terry and other researchers have done from 1988 till now. The Wheel of Excellence is a model that has been supported by many applied studies in extensive consulting experience with elite performance, and you can see in the reference in our research that many researchers have take this model as a theoretical framework. The statement of our research, because I have 3 minutes, 10 minutes? Okay. The purposes of our study were threefold. The first one was to identify the specific success element used by some high-level athletes in their pursuit of excellence. The second goal is to examine the degree to which Arab athletes were mentally prepared for the highest level of competition. And the third one is to offer an effective applied mental training program that consider the specific needs and mental skills of Arab athletes as well their respective context. The significance and the importance of this study wishes to bring first a new information in the field of Arab excellence. Second, to provide Arab coaches, athletes, and sport administrators a view of how Arab top athletes perceive the elements that lead to excellence. And the third, to provide Arab athlete and coach and mental trainer and also all the person who are in charge of sport in the Arab countries with a mental plan for improving the performance of Arab athletes on the world stage. And this mental plan was elaborated by Terry and I, and we need to now make it in practice with some athletes in different countries. In terms of the methodology used, the participants was Arab athletes, Arab coach, and sports psychology consultant from Morocco, Qatar, Tunisia, Egypt, Saudi Arabia, United Arab Emirates, and Qatar. The instrument used was an approach which was a triangulated approach combining qualitative and quantitative instruments. The quantitative instrument was the interviews with athletes and a survey that we have made for the Arab coach and sports psychology consultants. And in terms of the quantitative approach, we have used a questionnaire called the OMSAT, which is in Arabic and in French. The quantitative instrument was the Ottawa Mental Skill Assessment Tool, and the qualitative instrument was in-depth interviews and our open survey questionnaire on mental training. Just some details about the two instruments we have used. About the open survey questionnaire on mental training, we have asked the coaches and sports psychologists from the Arab countries about some knowledge regarding the mental training preparation of the higher level athletes of their respective countries, and to ask them about the importance attached by their respective athlete to mental trainings, and to ask them about the existence of a mental training program devoted to mental preparation. In terms of the Ottawa mental training, mental skill assessment, the OMSAT, it is a tool that assesses a broad range of mental skills. It's, they have 12 scales regrouped into 3 categories, and each categories you have foundation skills with goal settings, self-confidence, commitment, and psychosomatic skills. It assesses some psychosomatic skills regarding fear control, relaxation, activation, focusing, refocusing, and some cognitive skills as imagery, mental practice, competition planning. There you have a result from the OMSAT regarding 2 populations from Morocco and from the Qatari athletes. And we can note in this table that the commitment was the first element that determines or leads these athletes to excellence, followed by confidence, and the focus is in 3rd place. But the element that was a surprise for us is the ongoing learning, and I will ask Terry at the end of this presentation to talk about this problem of ongoing learning and compare it with Western athletes. For athletes in the study, commitment was identified as one of the major determinants of excellence and the achievement of their goals. Their commitment was also evidenced by some aspects as dedication, sacrifice, resistance to intense training, willingness, perseverance, patience, and responsibility. And here I have some meaning units that confirms this commitment and sacrifice. For a woman athlete to reach international level, she has to sacrifice several feminine aspects, among others, its hair, its feminine size. She has to forget all these coquette clothing aspects. I would mean that Moroccan women athletes cannot access to international level without sacrifice. That was a woman world champion. Sometimes for another athlete from Morocco too, sometimes when I leave my room in the training center, I made my goodbyes to my friend. I was determined to train to that. That means that this athlete can go through anything to reach their aim. In the majority of our interview, we have noted that the love of the sport and the love of the discipline have chosen, the discipline chosen was a reference for all these athletes. They felt that this love provided an element of motivation that guided their pursuit, persistence, and success of their performance. I go. Here you have something about concentration, imagery, and distraction control. Moroccan athletes as well as Qataris have difficulties managing fear. That's a great problem with fear and refocusing. They have developed their own. We have in my interviews, I have found that they have developed some strategies that they have developed own selves to divide from the difficulties and all this distraction that can be internal or external by reading some verse of Quran to better concentrate. Their confidence seemed to give them a moral support to overcome situations which can affect their performance. Moroccan and Qatari athletes seem to be aware of the importance of the mental imagery for the excellence in sport. This mental strategy helps them to repeat the race or the skill mentally to get ready to the competition. Regarding the perceived mental skills associated with sport excellence among Moroccan and Qatari athletes, the results indicate clearly that major performance blocks or setback that interfere with Arab athletes was a lack of focus, an inability to control distraction and stress, and the absence of an effective process for evaluating and drawing out the important focus lesson from their experience. The finding of this study indicate clearly that Moroccan and Qatari athletes associate several mental elements to excellence in sport. For this athlete, commitment, confidence, distraction control, and positive image seem to be the major factor that determine the athlete's success. Regarding the survey that we have done with sports psychologists and coaches to examine their knowledge regarding the mental training preparation of high-level athletes, Arab coaches as well as consultants seem to recognize the importance of the mental training and effective focusing training in optimizing performance. Second, we have noted that they were strongly concerned about the absence of meaningful applied mental training in the general preparation of their respective athletes. All of them were aware of this absence of the mental training program. However, they emphasized that it is time for Arab sport officials to consider this critically important issue and start to integrate it into the general preparation of the athlete. Here are some meaning util. Today, I go through the. When key officials were asked about the deficiency of mental preparation and general preparation of the athlete, the majority of respondents deplored the absence of Arab sports psychologists, but justified that by lack of time, lack of knowledge, and unawareness of this aspect. Here we have a mini unit, very interested. To be prepared for the Olympic is not an easy task. The athlete should be prepared physically and principally mentally to overcome the pressure of the event. This requires multidisciplinary work supervised for several years by a team of experts in the field of sports science. The recommendation that we have got from this survey that mental training or focus training for excellence is clearly a necessity for consistent high level of performance, especially in major international events. Arab athletes should be introduced to a high quality mental training preparation program as part of their normal training. And they insist on the need of an effective and applied mental training program that can be integrated into the general preparation of the athletes. I go now to the conclusion. To the goal of the Arab focused training plan is the plan we have made is to ensure that Arab athletes are fully mentally prepared for their most important high-level competition, are focused on the right things at the right time, that are clearly understand what kind of focus freedom to perform their best and what kind of focus interfere with their best performance, are well prepared mentally to perform their best and deal effectively with distraction at high level performance, and are encouraged to train with a positive and fully connected focus. Here we have some tools, and I go to the conclusions that conducting this collaborative applied research study on performance excellence with the intent to help Arab athletes become the best performance they can be. It was a very cooperative and collaborative mission from start to finish, and everyone grew from this process. Arab athletes certainly have the potential to be among the best in the world on a consistent basis. Our goal and recommendation are directed at helping these Arab athletes turn their dreams into reality and helping them turn their dreams into positive reality. A very important point to keep in mind when pursuing positive dreams is to remember that only action counts. Thinking is not enough. Talking is not enough. Even deciding is not enough. We have to act now, individually and collectively, on our intention to turn positive dream into positive reality. And we wish that this quest, hope that this modest contribution will help all Arab athletes, coaches, and sports psychologists in some meaningful way. And shukran, thank you very much.
شكرا أستاذ دكتور بريع على هذا العرض المختصر، وأقول مختصر لأن البحث ما شاء الله في أكثر من 120 صفحة، وبالتالي كان ضغط على الدكتور عشان يتمم هذه المحاضرة. لإتاحة الفرصة للدكتور نيج لعرض بحثه أو عرضه الثالث، فليس أمامنا إلا 5 دقائق لطرح سؤال أو سؤالين على الأكثر حتى لا نأخذ كثير من وقتكم. فالحضور كريم يتفضل. مايك بليز. وأتمنى من الإخوة طرح السؤال بإيجاز إذا تكرمتم.
السلام عليكم. أستاذ دكتور عبد الرزاق عبد الجبار حمزة. حقيقة سؤالي إلى الأستاذ الفاضل دكتور عبد الرحيم حول مسألة أكد على الجانب النفسي، على التدريب الذهني. أنه هو الذي وضح أنه هو الأساس في تحقيق الإنجاز، تحقيق المستويات العليا. إلا أنه فات أن يذكر هناك علوم أخرى أكثر أهمية أو تقف إلى جانب هذا علم النفس في عملية تطوير الإنجاز الرياضي في الوطن العربي. ولذلك نشوف يعني تخلفنا في الجانب التكنولوجي، في جانب الفسيولوجي، العلوم الأخرى اللي كلها تدخل مجتمعة في تطوير عملية التدريب الرياضي. نشوف إحنا الميداليات اللي ذكرها اللي حققوها العرب طيلة الأولمبياد هي 105 تقريبا، أكثر من 40% في مجال ألعاب القوة، واللي يريد أقوله أوضح أكثر في مجال الجري، عدا يمكن غادة شعاع كانت تحتاج إلى تكنيك. يعني نشوف إحنا متخلفين في الألعاب اللي بيها التكنيك عالي، لأن لا يوجد عندنا تطور تكنولوجي في هذا المجال، في مجال التدريب الرياضي. نشوف عندنا بالفترة الأخيرة كانت أيضا نوال متوكل في 400 متر حواجز، أيضا تحتاج إلى تكنيك. وأيضا آخر شيء البطل برشم، أسامة برشم في الوثب العالي اللي يعتبر الآن هو بطل العالي، أيضا تكنيك عالي تحتاج. فشوف تخلفنا العرب، أعز أنا إلى تخلفنا في الجانب التكنولوجي، ولذلك شوف التكنيك عندنا الألعاب اللي بيها تكنيك عالي شوف إحنا متخلفين فيها.
شكرا دكتور، تفضل. فقط شنو عندك؟ ما عندكش؟ خذ مايك، خذ لا هات مايك، لا مفتوح، افتح فيجن فينا تحت الجرين، الجرين اوكي.
الو
تفضل.
تسمعني؟ شكرا على السؤال الوجيه والمهم. يمكن الإجابة تكون باللغة العربية، ما في مشكل، لأنه طرحت السؤال باللغة العربية سأحاول الجواب باللغة العربية. هو كما قلتم كما تفضلتم، هو واضح بأن الميدانيات التي حصل عليها العرب من أمستردام للندن كانت أكثرها في ألعاب القوى. إلى رجعت إلى البحث أو اللي فيه تدقيق بجميع الميدانيات التي حصل عليها العرب، هناك في البحث جميع الرياضات التي فازت، ومن بينها الرياضات الألعاب القوى التي فازت بكثير، تقريبا أكثر من 60% من هذه الميدانيات 105 تماما. هناك كما تفضلتم من ناحية الإعداد الرياضي في الرياضات العربية لا زال مهملا جدا، ولكن مع الأسف الآن الكل واعي بمدى أهمية هذا الجانب اللي هو يكمل الجوانب الأخرى، لأنه الجوانب الأساسية للرياضة كما تعلمون فهي جوانب تقنية، فيزيولوجية وصحية والكل هذا الجوانب لها دخل ولها، وهي تتكون من ناحية systemic. It's a systemic approach where you have all these determinants, all has its importance on it. The performance is not only physical or technical or tactical. It is more this for all these components.
أشكركم على هذا الجواب المختصر، ونتيح الفرصة للدكتور نيج والدكتور عبد الرحمن العنقري للتفضل إلى المنصة لتقديم العرض الأخير. والدكتور تيري أورليك والدكتور بريع رهن إشارتكم لطرح أي سؤال بعد الجلسة، لأنه احتراما للوقت سأنهي هذه الجلسة فيما يتعلق بجانب الجوانب المتعلقة بعلم النفس الرياضي. شكرا لكم.
بسم الله الرحمن الرحيم والصلاة والسلام على نبينا محمد وعلى آله وصحبه أجمعين. I'm very pleased and honored to be chairing this session with Professor Berneg, and I'll give you the history of Professor Berneg in brief. So he started his bachelor degree in nuclear physics in Switzerland, and then at the graduate school he's shifted to biomechanics in general, and then moved to Canada. Started working in University of Calgary in biomechanics, of course, and he has published more than 360 scientific publications and authored and co-authored more than 13 books. And he's still working in sports biomechanics in University of Calgary now, and he's going to be presenting his scientific accomplishment in the field of sports biomechanics, and I think he's giving about 40 minutes to give his presentations. So hopefully he's going to stick to maybe 30 minutes, because maybe everyone is a little bit tired. So we're going to leave 10 minutes for questioning, if there are questions, or we're going to end the session. Thank you very much. So I'll invite Professor Neg to start his presentation.
I would like to talk about energy and performance. And I realize that energy is used in many different ways. When I talk about energy, I talk about the ability to do work, and I talk about different forms of energy, but I concentrate on the mechanical energy, knowing that there are other forms of energy. When we want to use energy to improve performance, when we want to use energy to improve performance, we have basically three possibilities. One is to store and return energy, like the diver here. One is to optimize the system, and one is to minimize the loss. And I would like to discuss these three possibilities quickly. First, storage and return of energy. In principle, you have a mass, you have a spring, you stretch that spring, therefore you load the spring, and you can get the energy back when the spring shortens. The energy is proportional to the spring constant and to the deformation squared. So the deformation becomes more important the bigger it is. And when you load a spring, you store energy. The area under the curve is the stored energy when you have the force deformation diagram. And when you unload, you go back and you have a difference between the loading and the unloading curve, which is called the hysteresis. And that difference is basically the energy that you have lost due to friction, to heat, energy dissipation, and so on. So when you want to store and return energy, deformation is the important part. And if you take a muscle, for instance, a muscle tendon unit, then you stretch that muscle tendon unit, and that deformation means you store energy. We know that we have some examples that are fantastic. Pole vaulting, for instance. Before changing to the fiber elastic poles, the world record was about at 480 4 meters and 80 centimeters. Now it's over 6 meters. In diving, you have the diving board. The trampoline, you can get fantastic altitude. The floor exercise, you couldn't jump triple somersaults without these gymnastics floors. In the tumbling, it's the same. So when you want to have energy return that is really meaning something, it must be substantial enough, must be at the right time, the right frequency, the right location, and the right direction. Let me explain that. In principle, when you have the elongation here and the time, you need the first half of the deformation to deform, and the second half to get the energy back. Now, the problem is that you don't have such nice curves. In running, for instance, you have two peaks. An initial peak, which is called the impact peak, and a second peak, which is called the active peak. And it is obvious that this impact peak has a different frequency than the active peak. The impact peak has a frequency which is between 10 and 20 hertz, while the active peak has a frequency which is about 1 to 2 hertz. So if you get something back here, it's with the wrong frequency and at the wrong time. So the deformation of the heel cannot be used typically to store and return energy. Only the deformation of the forefoot can be used. So when you see a shoe like that, you have this part here more for comfort and this part for energy return. And as a matter of fact, this shoe had an energy return of about 2.6%, which is quite substantial. The right direction. Now, energy is a scalar, but energy that is returned produces a force, and that force has a direction. And that direction can be used for performance. If you load something, you run in this direction, so from right to left, you see how these elements are deformed, and they return coming back in this direction. So you can build shoes or surfaces that have a direction. So it is quite clear that we can do something with respect to equipment. Can we do something with respect to the human body? We know that the kangaroo and the horse have long tendons, and it is thought that the kangaroo and the horse have fantastic situations for energy return in tendon. Here, the kangaroo, that's the speed, and this is the oxygen consumption. And you see that with increasing speed, the kangaroo does not increase the need of oxygen, which is surprising. But this is related to the fact that we have some resonance. And you remember the famous sprinter Oscar Pistorius, who was running 46.9 with 2 artificial legs. Here, an example in his run in Rome at the meeting. And the question was at that time, is he allowed to compete against normal body athletes? Now see what he does. He's on the outside lane, so that means he should always be ahead of everybody. Now he is already number 2, and here already number 3. Here he is, now number 4. So everybody passes him in the first 200 meters. And then in the second curve, see what he does. Here he comes. Look at the distance between him, and look what he does. And if the race would have been a little bit longer, he would have won the race. Why that? Yeah, IAAF, they made a court case out of that, and they said Oscar Pistorius, with his current prosthesis, has an unfair advantage over able-bodied athletes. And there were 2 biomechanists on each side. The biomechanist that said he has an unfair advantage, and the biomechanist that said he doesn't. Now, let's try to understand what's going on in the human body. The results that have been published is that in the Achilles tendon, one stores per step 35 joules. In the arch, and these were both Nature papers, in the arch, one stores 17 joules. So in total, one stores 52 joules. Now, the mechanical energy per step is about 100 joules. So that means that about 50% of the mechanical energy that is needed is returned. That's huge. That's unbelievably huge. Well, let's look at that critically. If that would be true, that means that all the top athletes would have the same arch stiffness, which is not true. It would mean that athletes that run with stiff orthotics would have a bad performance, which is not true. So I think one can forget the arch part. And the same is true, one has a lot of reasons to say it for the energy with the tendon. As a matter of fact, the simple thing is, if we humans would return 50% of the mechanical energy, we would hop 100 meters. We would not run 100 meters. So the Court of Arbitration ruled that the IAAF had not provided sufficient evidence to prove an advantage over able-bodied athletes. So in other words, we cannot really determine how much energy is returned. Or in other words, storage and return of energy in humans is not well understood for the improvement of performance, and the current numbers, I think, and suggest, need revisions. We don't return 50% of the mechanical energy. At the right time, at the right frequency, at the right location. Let me show you a little idea how one could store energy. This is a disc, it's called the power disc, that we developed together with an inventor, that is in the heel of the shoe. And when we have a typical control situation, then we have the typical force information diagram that looks like that, and the power disc has a different one. So when we deform the heel of the shoe, the deformation comes back, and when we lift the heel, the deformation is basically back. That means we don't get that energy back for performance. However, when we take that power disc, you see that the deformation is kept, and when the heel lifts, we get the energy back. We have measured in pilot studies the result, and we had for walking 3.7% less VO2. So there is a possibility in shoe to do that. or in general terms, storage and return of energy is large for some equipment. It's limited for shoes. We may be able to do something, but it's not that big. It's quite large for some surfaces. It's large for selected animal, and it's certainly contradictory for humans. So far, storage and return of energy. The second idea is to optimize the system. You all know that there is something like a force length and the force velocity relationship for muscles. We have learned that. Most of us have learned that. So if this is the force length relationship, this is the contraction velocity, this is the force produced, then we know that the faster we contract, the less force we can produce. Now, force times velocity is the power, so this is this curve here. That means somewhere at a certain velocity, we have an optimum. Or in other words, if we have a certain muscle and we want to produce the maximal power, we can do that at a certain velocity of contraction. Now, we did some studies. A colleague of mine, Dr. Herzog, did a study where he did that for cycling. And he built a model where he could change the pelvic inclination, the seat height, and the rate of crank rotation. Let's look to the result for the rate of crank rotation. These are all the different muscle power curves for these muscles that are here indicated. And this is the resultant curve. Or in other words, we have a resultant optimal crank rotation for maximal power output. That means we can optimize the system. We can do that for a speed skater, we can do that for a cyclist, we can do that for many sports. It is not often done, but I think it would have a lot of possibilities. Cycling for sure. The last point is minimize the loss. The idea is, if I walk here, I use a certain amount of energy. If I would walk on a mountain, on the top where it is like that, I would spend about half of my energy to make sure that I don't fall down. Or in other words, I have some energy that I use to do the task at hand, and some energy that is not task related. And the energy that is not task related, we want to minimize. So, for instance, friction, low hysteresis, or minimize muscle work to avoid soft tissue wobbling. If I walk like that, my soft tissue wobbles, and I have to spend energy to damp those vibrations. You have that in skiing. But I would like to talk about one piece of work that we did, my colleague Darren Stefanishin, in his thesis with me. He had a result that when you do a movement, you bend the knee. This is this part. You bend the knees, the energy is absorbed. You stretch, energy is generated. And every joint has that. The hip joint, the knee joint, the ankle joint, except the metatarsophalangeal joint. In running, the metatarsophalangeal joint has only energy absorbed, but not energy generated. So that means we lose energy, but we don't produce energy. So the idea was to make stiff shoe soles, and these stiff shoe soles would have a better performance. Here, the metatarsal power, normal, stiff, very stiff. Therefore, we would expect a better performance here than here. The result here for one athlete, you see the normal spikes, stiffer, stiffer, and you see the improvement is about 2.4%. And we did that with 32 athletes at the top level, all better than 10.8 over 100 meters. And look at that. We had 3 athletes that did not have an improvement. All the other ones had an improvement, and the improvement was in the average 1.7%. Now 1.7% in 100 meters, that is almost 2 meters. The interesting thing is that it is not the stiffer, the better. There's somewhere an optimum, because the result depends not only on the stiffness, but also depends on the strength of the triceps surae. But all the middle distance running shoes have stiffer soles now. Another example is soft tissue vibrations. I said every step you have a shock. The shock means that you have vibration of the soft tissue, and you have to damp those vibrations that cost you energy. Now, if you have a compression suit that has the right properties, viscoelastic properties, then you can damp the muscle vibrations, and you don't have to do the muscle tuning yourself, but the apparel could do it. Now we did that with the test suit here, and here the control suit, and the improvement was 1.7%. As a matter of fact, this suit here was used by the winner of the World Championship 2001 in Paris, over 100 meters, a person that won and was not expected to win, because he had certainly an advantage because of that suit. So minimizing the loss of energy has lots of application: stiff soles, soft tissue vibration, stability. And when we try to summarize what we learned in that almost half an hour, first, we have 3 strategies possible: storage and return of energy, optimize the system, minimize the loss of energy. These are the 3 possibilities. Energy storage and return can be very high in equipment. The return in the human body, especially the Achilles tendon, is not clear. I would suggest it's small. And the return in the foot, again, I would suggest is small. So storage and return of energy, primarily a thing for equipment. Optimizing the system has some possible application. It's not done at the moment in high-performance sport, but it should be done. And minimizing the loss of energy is probably the best strategy at the moment that we have, and I think we can produce quite good results with that. So, if you do that, you can have surprising results. You probably haven't seen it. Thank you.
Dr. Neg, for this informative and enlightening presentation. I think we have some time for 1 or 2 questions at max. And if somebody wants to ask a question, no one is going to ask, then we have to end the session. Okay, well, thank you very much, Dr. Neg.
شكرا جزيلا للسيد رئيس الجلسة والمتحدث الكريم. شكرا لكم سيداتي وسادتي على حضوركم. نأمل إن شاء الله أن نلتقي بكم مساء. التجمع الساعة 6:30 مساء في هذه القاعة، والحفل يبدأ إن شاء الله الساعة 7 مساء. قبل هذا نرجو قبول دعوتنا على الغداء في قاعة بنياس 3، القاعة المجاورة. فشكرا لكم ونلتقيكم على الغداء إن شاء الله.